new insights in the development of novel biofertilizers and biopesticides. So this is the workshop 13, which is going to organize on today on 12 October only. And this is the part. This workshop is the part of 21st century, the era of biotechnology. This is one refresher course we are organizing since 3rd September, and it will finish on 21st October of this month. So I heartily welcome all our distinguished speakers of this particular workshop. And this particular workshop, firstly, we have a keynote talk, and that will be delivered by Dr. R.C. Dubey sir, who is working as Professor and Dean Faculty of Life Science, Gurukul Kangdi University, Haridwar. After that, we will attend the lecture of Dr. Aradhna Mishra. She is working as Principal Scientist. Dr. Mahavir Sharma, he is working as Principal Scientist at ICR, Indian Institute of Soybean Research at Indore. Then our next speaker will be Dr. Ramakrishna Burista. He is Professor at Department of Biochemistry in Central University, Punjab. So, one of the speaker is not presenting today because he is having some meeting in Delhi. So he just requested that please arrange my lecture on tomorrow in the morning. So on tomorrow, Dr. Bhim Pratap Singh, he is working as Associate Professor at National Institute of Food Technology and Entrepreneurship Management at in Haryana, Sonipat. So he is going to present his talk on tomorrow. So overall, now we are welcoming our guest, and firstly, Professor R.C. Dubey sir will deliver keynote talk. So before starting this particular workshop, I firstly invite our convener, Dr. Professor Anil Prakash sir from Barkatullah University, Bhopal, because he is chairing whole workshop as convener. So before starting this workshop, I just request Anil sir to say a few words and to start this session. Dr. Anil Prakash sir. Anil sir. So there might be some audio related problem. So we should start because Dr. Anil sir already told us that please start the session because speakers are present here. So firstly, I would like to invite Professor R.C. Dubey sir to start this session as his keynote talk. Before starting this talk, I just give a brief introduction of Professor R.C. Dubey sir. Professor R.C. Dubey sir is a professor in Department of Botany and Microbiology in Gurukul Kangdi University, Haridwar. He has earned his MSc and PhD degree from Banaras Hindu University in the year 1981 and 1986. And he served in Kumaon University as lecturer. And after that, in the year 1997, he has joined Gurukul Kangdi University as reader. And after that, he has become professor in the year 2004. Since that, he is working as various capacity in Gurukul Kangdi University, and his field of interest in rhizosphere microbiology and phytopathology, and having expertise in biocontrol of soil-borne plant pathogens, mycorrhizae, rhizosphere microbiology, mycorrhization helping bacteria, microbial biotechnology, and botanical pesticides. He has published more than 150 research papers, review papers in national and international journals of repute, and he has produced more than 25 PhD on various aspects of microbiology. And I am having the honor that I am one of the student of Professor R.C. Dubey sir. I have completed PhD under his kind guidance in the year 2008. He has handled research projects sponsored by UGC and CSIR. Professor Dubey has been a member of Research Degree Committee and Board of Studies of several universities and he has attended dozens of national and international seminars, presented research papers, and delivered lectures. Professor Dubey has authored more than 5 books on microbiology, and one of the famous book, that is textbook of microbiology. He is the one of the first author of Indian origin who has written a book on textbook of biotechnology. And also he has published practical microbiology, advanced biotechnology books, and he is also co-editor of 8 books of different uh, editions from different publishers. He has also published a compilation of his poems in the form of book of XIII in the year 2014, and he is the original organizing secretary of many national and international seminars and workshops that were funded by many funding agencies. He was coordinator of many workshops also. So Professor Dubey is life member and fellow of Indian Botanical Society, Indian Phytopathological Society, International Society for Conservation and Natural Resources, and he was associate editor of Journal of Indian Botanical Society and member of editorial board of Journal of Environmental Biology and Conservation, and he is nominated as Counselor of Indian Botanical Society for 3 years. So at present, Professor Dubey sir is working as Dean in Faculty of Life Sciences and also Faculty of Medical Sciences and Health in Gurukul Kangdi University, Haridwar. So now, with these words, I am handing over. I am this particular workshop speaker and to Professor R.C. Dubey sir to deliver his keynote talk and enlighten us with the particular area that is related with biopesticide and biofertilizer, so that we can learn new insights in this particular work from his expertise. So now I am welcoming Professor R.C. Dubey sir. And before starting this workshop, now I am welcoming Professor Anil Prakash sir, who is here as a workshop convener. Anil sir, please, your words.
Thank you very much. I welcome Professor Dubey. I am audible?
Yeah, yes sir. Yeah.
Hello. So first of all, welcome the Professor R.C. Dubey for the keynote lecture. So I will not waste much of time. I just request Dr. Dubey to move forward to just give your. Thank you very much. Professor Dubey.
Yes.
Thank you, Professor Anil Prakash and Dr. Kamlesh Chaure and your team for inviting me to deliver a lecture in the refresher course on the topic of novel biofertilizers and biopesticides. I have selected the topic biopesticides for sustainable agriculture. We know that agriculture is one of the most greatest demand of the life of human being, because agricultural practices give us the food, and to animals the feed, to microorganisms the nutrients. So without agriculture our life is not possible. It has been mentioned as kalau annagata prana loka solpayu khastata. The prana means the soul resides only because of the nutrients. If there is no nutrient, no life. So nutrients are the life giver. As far as this practice is concerned, I want to draw your attention about more than 10,000 years BC, when during Neolithic age the persons is starting agriculture. In the Rig Veda also, the practices of agriculture has been described as methods of increasing the agricultural products by using the dungs of goat, sheep, cow, water, as well as meat has also been described. During Mahabharata period, more than 6500 BC, rotting corpse or stinking garbage returned to the earth are transferred into wholesome things that nourish life. This statement has given, and in Mahabharata as well, there is a mention of several manures oil cake, animal excreta that upon aiding the soil increases crop yield. My dear friend, you know that with the pace of the increasing, with the pace of increasing in the number of individuals day by day in the on the earth surface, the demand of the nutrients is also increasing. When India became uh, independent in 1947, the total population was approximately 37 crore. At a gap of 70 years, the number has gone just to 3 times more. 135 crore population is of Indians. In the same way, in the world also, there was time and population was about 350 crore and the number has just doubled. Where from this the food product will come? The area and size of the field is shrinking, but the number of eaters of the food is increasing day by day. So still it needs more attention by the scientists. You know, always the persons were suffering from food shortage, and since the civilization. I'm sorry, somebody is talking to me.
No, sir, you can carry on. Professor
Okay.
RC sir, your
Okay.
slide is not visible, sir.
But I have shared again. I am just again going. Please, I am again sharing the slide. Please wait. Is it visible?
Yeah. Yes, sir. Yes, sir. It's visible, sir. Can continue.
May I go ahead? May I go ahead?
Yes, sir. Yes, sir.
So you know that in during 1974, it was Borman Norlog who coined a term Green Revolution, and he was awarded with Nobel Prize. You know, the Green Revolution was possible only because of addition of the chemical pesticide and chemical fertilizers. And today's scenario is that all throughout India in 2020, there is a total consumption of the pesticide for about 54,121 tons. And more than 50% of these pesticides are used in the cotton and about 18% to the paddy. My dear friends, do you know where these pesticides go? Ultimately, it enters in the it enters in the soil. It enters in the plant. Anyway, there are alternatives to increase the crop production as well as crop protection by opting these two methods, either crop production or crop protection. We can enhance the total amount of food grains available to our consumers. Crop production has been possible because of opting organic farming, green manuring, chemical fertilizer, and biofertilizer nowadays, and crop protection by adopting chemical pesticide as biopesticide. So these two, biofertilizers and biopesticide, needs needs far more attention in near future because of several reasons. Because they pose no side effect. I want to give some cases in United States. 4 lakh farm workers are affected by pesticide related illness. such as dizziness, vomiting, and severe skin rashes. More than 8 lakh people fall ill due to pesticide poisoning, and about 14,000 thousands of them die in agony. The number of cancer cases, neurological problem, reproductive effect, and fertility is also being affected by the use of these fertilizers in India. There is a case of Chandigarh. People have find out that the workers who apply the pesticide in the field, the chromosome number of the sperms become duplicated. So this is a most dangerous effect. I want to just memorize you Bhopal gas tragedy of 1984. This is the saddest industrial tragedy of 20th century in India, where in the night of 2nd December 1984, the MIC factory, methyl isocyanate, it was produced in a industry, and the gas got leaked up. More than 80,000 persons died in the same night. According to unofficial report, according to official report, only 8,000 people died. There is a question not of the number, but the question is to the death of the person. The industrial tragedy only due to because of leakage of methyl isocyanate that kill such a number of individuals is very tragic one. Now you can see some of the pictures of that days where people lost their life. The survivors who are still 1,20,000 victims are still suffering from side effect, and the main culprit of the methyl isocyanate industry was Warren Anderson, and Union Carbide chief Warren Anderson was arrested, and on the same day he was released and went to America. He did not turn up again, and no compensation was given by him. My dear friends, after a long gap, still we are using the pesticide not in calculated amount, but in free hand. You can just see in some of the pictures, there is no any ratio of mixing the pesticide in the water. In a free hand, these pesticides are sprayed in the paddy and in some other vegetables. Day by day, we hear in the news, in the telecast in television, that the number of cancer cases are increasing in India only due to this misuse of the pesticide. Now in India, this green revolution was made possible mainly in the Punjab region. In idea, about 1,300 cancer patient per lakh of population. So there are several drawbacks of agrochemicals. It develops microbial resistance, imbalanced soil nutrient, residual impact in food chain is caused by it, groundwater pollution is done, the high cost and economic loss occurs due to pesticide, and mismanaged agroecology is resulted. Now this is a picture of the state Punjab, where in different states the situation of cancer percentage too high. This is alarming. And one train goes from Bhatinda to Bikaner, from Punjab to Rajasthan. The name of train is Cancer Express. The name of this train is Cancer Express because the cost of treatment in Bikaner is low as compared to that in Bhatinda. So number of travelers in this train is maximum. Now the pesticide uses per hectare agriculture land, this is a scene. Maximum users is South Korea, Malaysia, Pakistan. In India is also using this such type of pesticide, but less area is used by Indians. Now in India, the West Bengal uses 5%, other state 15%, Andhra Pradesh, Telangana 24%, Punjab 11%, Karnataka 7%. So this is a picture. Maharashtra 13%, Tamil Nadu 5%. Now, in India, fertilizer market production of diammonium phosphate and complex fertilizer in terms of metric ton in 17-18 was also high. This is complex one, and day by day the use is increasing. Just keep in mind the size of the land is decreasing, but the use of fertilizer is increasing. Now, what are the alternatives? Alternative is use the biofertilizer and biopesticide. The biopesticides are a form of pesticide based on microorganisms or natural products. The biopesticide work through different mechanisms. For example, amensalism, that is antibiosis and commensalism, parasitism, which includes mycoparasitism, and antibiosis through the production of antibiotic and toxins. Now, as far as biopesticide is concerned, history goes to 17th century. Nicotine was used to control plum beetle. Then Beauveria bassiana was used in 1835 to demonstrate the control of insect pest of silkworm. Then 19th century, mineral oil was used as plant protectant. In 1910, Japanese biologists isolated Bacillus thuringiensis from disease silkworm. In early 1920s, the French began use of spores of Bacillus thuringiensis as biological insecticide. In 1938, sporine was made available in France as the first commercially available BT product. Now, during 1950, the widespread use of biopesticide began in the US to take hold as a host of research on BT efficiency. In 1956, Pacific Yield Product Company developed submerged fermentation, which produced Bacillus thuringiensis on a large scale. Then in 1973, Helionthes nuclear polyhedrosis virus, abbreviated as NPV, was used as the first viral insecticide called as Ilcar, and it was marketed in 1975. In 1999, kaolin clay, this is a clay mineral, which is also known as Multani Mitti. In Multani Mitti, there are several clay particles, clay minerals. One of that is kaolin or kaolinite. It was made commercially available for use in organic system as an insect repellent in organic fruit orchard. Now, why should we be focused to the biopesticide? Because biopesticides provide non-targeted effect. They are cost effective. They can be made available in very low cost. Biopesticides are eco-friendly. There is no damage to the ecology, no, in the environment. They are non-hazardous. Don't pose any hazardous effect like chemical fertilizer, and they work for longer time because microorganism can survive, can propagate, can multiply in the soil, and in this way they can work for a longer time. I will tell you later on the effect of this longer time. The biopesticide of different type, like microbial pesticide, plant incorporated pesticide, then biochemical pesticide. I will focus on microbial pesticide. The microbial pesticide market is gaining attention more and more. The sources microbial and biochemical. The grains, seed, fruit, and vegetables can be applied through soil spray, seed treatment, foliar spray, and the products available in the market are bioherbicide, biofungicide, bioinsecticide. There are different types like microbial pesticide, the plant-based biopesticide, biochemical pesticide. For example, microbial biosurfactant. The biopesticide market by region targeted to 2027 will increase circularly, but there is no every drastic or sudden increase of the green color, light blue color is the situation of Asian country. So in Asian country, this market will compared to 1927 will get a little momentum, but not so drastically. The biopesticide research and global patent is also increasing day by day. But as far as Indian condition is known, there is a very slow pace. This green color is of Indian content, and the orange color is of the global condition. So the number of patent and the production of the biopesticide is also gaining momentum, but to a slow speed. Worldwide biopesticide market, this is a compound annual growth rate. It is also increasing to in 12 to 17 by 15%, and expected to increase to up to 24 by 8.8%. Now, global market segment by region is this is a condition of India by 2025 as compared to other countries. This the production market would be parallel to that of Japan, but in Southeast Asia, it is expected that it will be maximum. So we have to focus more on this aspect. Now, trend of global pesticide vis-a-vis biopesticide market, I have discussed, nothing else. My dear friend, there is a large number of pests and pathogens that harm our agricultural crop as well as agricultural products. Needless to name these insect pests, I will give some of the examples. These pests can be categorized into three groups: Lepidoptera, Coleoptera, Diptera, and these are the pests. The caterpillar and the insects, they sit upon the plant leaves and stem, even fruit also. They chew upon, they make it perforations, and in this way they slow down, they degrade the quality as well as quantity, declines the quantity of the product. Now, for example, European corn borer feeding upon the corn, this is very dangerous. The number of corn users are too much throughout the whole world. So it is fed by the corn borers, and they destroy the quality. Now, different type of products can kill the insect pest, like plant extract, the bacterial products, like, for example, Bacillus thuringiensis or BT crystals or BT powder available in the market, the fungal preparation, the viral preparation, for example, nuclear polyhedrosis virus preparation available in the market. It kills the nematode as well as the insect, other type of insects. Now, in the viruses family, the most important is the Baculoviridae family, Baculoviridae, and on this work was done about 100 years ago, the baculoviruses, and they are an effective biocontrol agent of the insect pest. The baculoviruses have a distinct rod-shaped nucleocapsid of 30 to 60 nanometer diameter and 250 to 300 nanometer in length. There are two types, granuloviruses and nuclear polyhedrosis virus. So major difference between these two, that in granuloviruses, they are granular in appearance, and the nuclear polyhedrosis virus are crystalline in structure. And granuloviruses contain a single virion per inclusion body, and they have either single virion or multiple. They may be multiple. This is a picture of nuclear polyhedrosis virus. You can say single rod-shaped capsid of the virus, baculovirus. This is single rod-shaped capsid having DNA genetic material. This is a capsid, and it can multiply. It multiplies in the cells of the insect, and its replication occurs inside the nucleus. Because it is DNA, it enters directly through cytoplasm and goes to the nucleus. It multiplies large in number. After budding comes it out. Then uninfected cells will also be infected by the released particles. In this way, the number of this capsid is increasing, and ultimately the cells of the insects are degraded, and such insect will die. This is one of the best example of the nuclear polyhedrosis virus that kills Helicoverpa armigera, that eat upon the chickpea. This armigera that eat upon chickpea, it can be descended by nuclear polyhedrosis virus. If the same Helicoverpa infect upon the cotton, as known as cotton borer, it can also be killed by this nuclear polyhedrosis virus. Now this is other plant, the baculovirus. It makes perforations, and through the perforation it enters inside, and in the cell multiply large in number. In this way, in due course of time, you can say on the safe leaf, the insect dies. It cannot multiply larger in number, neither it can harm to the plant. Now the microbial biopesticide are throughout word-wise are use of the fungicide, fungal preparations as a bioactive ingredient, bacterial preparation, viral, and nematodes. 3% nematodes can be killed, 60% bacterial product, 3% of nematode being as used as nematode trapping fungi. Nematode trapping fungi, they can be killed. 27% fungal product and 10% of viral products are available. Now this is the picture of baculovirus preparation available in the market as baculovirus granular powder, and in this granular powder may be dissolved in the water, and it can be sprayed on the plant, or it can be sprayed directly mixing with some inert ingredient on foliar region. Now second example is Bacillus thuringiensis. The species are 5 different type. I am not going to say it's the species and its differentiation. Now in different species there is a presence of different type of genes known as XIII genes, earlier known as the Bt gene and Bt protein, now known as XIII gene, XIII 1 to 5, and these genes secrete the toxin protein, and the toxin protein eat upon different group of the insects and kill up. Now these plasmid, these genes are present on the plasmid. The proteins are produced, and these proteins can be prepared, multiplied a large in number in the form of crystal, or bacterial preparation can be produced and spread on the plant. The mode of action is such that it enters inside the midgut and disintegrate the cells of the midgut. Ultimately, insect dies. This is the crystal. The formulations can be done in the form of suspension concentrate, granular bait, ready to use bait, suspo emulsion, granular and oil miscible flowable concentrate, dispersible powder, and wettable powder. Different type of formulations can be made and can be applied to the plant. There are several soil-borne. I told you about the insect. Just switching over toward the soil-borne plant pathogens, there are large number of plant pathogens. They cause harm to the plant either in the field condition or in storage condition. Examples are Macrophylla facilina, Sclerotium ralpsi, Sclerotinia sclerotiarum, Rajectonia solani, Fusarium, different species of Fusarium, Pythium, Pseudomonas solanacearum, Agrobacterium thuringiensis. Now, symptom you can see, Professor Mehrotra is here. He is expert on working in Indore on soybean. I have also worked a lot on soybean. So this is a picture of soybean. You can say It causes charcoal rot disease on the soybean. This is the cauliflower. The roots, just you can see, Sclerotia sclerotineum. It has disintegrated the root, and ultimately in such plant fail to produce products, because these fungi can survive through its survival structure in soil for a longer period. Microfauna fasciculina, if present in tissues, it can survive for about 20 years. Keep in mind, Microfauna fasciculina, if enter inside the tissues, it can survive for about 20 years, causing infection on more than 500 plants, including the shrub as well as trees, herb shrub as well as trees. This is Rajactonia solani. Rajactonia solani is Sclerotium ralphsi. You can see here. This is a Fusarium chlamydospore. All these are Sclerotia, Sclerotinia, Sclerotiation. The survival period of these fungi varies accordingly. For example, Fusarium udum can survive for 3 years in the field, and similarly, the Sclerotium ralphsi can survive about 5 years in the soil. So survival period of these fungi vary. Type of bioinoculants or biopesticide can be prepared, on which I have worked. I am giving only my example. Antagonistic filamentous fungi, for example, Trichoderma harzianum is very successful, and products are available in the market. Similarly, plant growth promoting rhizobacteria, they are large in number. A large number of products are also available in the market. Then biosurfactant producing PGPR is also available in the market. Biosurfactant to be used in agriculture, or biosurfactant product producing PGPR and antagonistic bacteria as such. Now some pictures of Trichoderma harzianum. This is our publication. It kills, colonizes and disintegrates the hyphae of Microfauna fasciculina. The quorum sensing molecules are produced by by the plants as well as by the antagonistic and the rhizobacteria. It has been done by MI ISDEN, and through these quorum sensing molecules, these bacteria colonize the root system very easily and warrant the entry of the pathogen. One of it is acyl homoserine lactone, used as the quorum sensing molecule. The biosurfactant properties are that these molecules have both hydrophobic and hydrophilic domains. They are capable of lowering out of surface tension and beneficial tension on the growth medium, and they have great importance due to their structural and functional diversity for agriculture application. Now, the biosurfactant producing PGPR are biocontrol of pathogens used for pesticide in industry, biofuel formation, in plant growth enhancement, as well as for soil bioremediation. Now, this is the screening method of biosurfactant producing rhizobacteria. You can screen out in laboratory. Our students are doing on biosurfactant producing rhizobacteria, and you can identify through chemical method, by FTIR spectroscopy. We have done on Enterococcus species, through GCMS spectra of purified biosurfactant. You can identify. We have found that glycolipid type of biosurfactant was found in Enterococcus species, and it was, you can see here this is Enterococcus species in dual culture. It is restricting the growth of microfauna. In this case, only biosurfactant is there, no live bacterial cell. It is also doing. In electron microscopy, you can see this is the bacterial cell that disintegrate the mycelium, and this is the biosurfactant itself. This is chemical, not the cell that disintegrate microfauna. In electron microscopy, this is very clear, and such hyphae loses its structural integrity, cannot survive for a longer time. Otherwise, it survives for about 20 years after. I told you this is publication of my incident, biosurfactant mediated biocontrol of microphomina facilina causing charcoal rot vigna. Now, growth inhibition by Pseudomonas, fluorescent Pseudomonas, GRC2, and different aspects we have done. Electron microscopy, you can see here the perforations made in sclerotia that is responsible to survive for 20 years. Here you can see the perforations made in the hyphae, and such hyphae cannot survive. Otherwise, it can survive for longer period. These are sclerotia. Sclerotia are being killed by other species of the bioinoculant, Azotobacter cruococcum. This is a free-living nitrogen fixture. It also acted besides being as the biofertilizer, bioinoculant. It can also be used as biopesticide because it kills the microphomina and other type of pathogen. This is the work of my daughter, who also found the bradyrhizobium. At one place, I said biofertilizer, but on other time, the same bacterium acts as biopesticide. It also kills this survivor, they kills the pathogen, microphomina, disintegrates the hyphae, and declines the survival of microphomina. In this way, crop can be protected. My dear friend, now these bioinoculants can be multiplied on large scale using fermenter. You can produce bioinoculant on large scale, and these bioinoculants can be used in agriculture. We have done on mass scale in the agricultural field such type of work for mass application, mass production. And this is a laboratory experiment where you can see the mixture of bacillus to bacillus species. It along with microphomina facilina, you can say it can protect and increase the growth of the vigna mugo, and if microphomina alone is there, it causes disease and production will be low. In field condition, we applied on system Azotobacter cruococcum. In the first year, there was enhancement in production. Also, without using any fertilizer, the production of disease. Now you can see the other crop, sunflower. Sunflower was applied with Pseudomonas species, and in all the cases there was enhancement in the production being without using any type of fertilizer. My dear friend, in another experiment, we applied the Pseudomonas aeruginosa GRC strain in a field in 2000. Subsequently, in the same field, without adding any fertilizer, the paddy was used in second year. In third year, again paddy was used. There was no application of any fertilizer or biofertilizer. It was usually once, first year in 2000, but production was continuously increasing. Compared to first year, in third year also, there was no application of fertilizer or biofertilizer. The biofertilizer was used only in first year, but the production continuously used in 2000 to 3 years also. So we published a paper in current science, long-term effect of Pseudomonas aeruginosa on the yield of subsequent crops of paddy after mustard seed bacterization. So the application was done only once. Production was done for 3 years consecutively without using any type of biofertilizer or fertilizer in the field. This is the result of field application. There are, as I told you, several commercial products of the biopesticide are available in the market. This is a neem-based biopesticide, neem-based. This is biotechnology, biocare, the microsakti. This is the name of any firm. The Prabha Dharma, produced from Allahabad. Then Azo Zero, where Azotobacter is there. These are some of the examples only. A large number of products are available in the market, and commercial. Commercially, these products are being produced by different pharmaceutical different agricultural company like Plant Rich in Kerala, Scientific Agricultural Laboratory in Madurai, then High Tech Agri Science in Bangalore, Central Biotech Nagpur, Innova Agro Laboratory Hyderabad, Sri Krishna Biotech Industry in Betul, Greenway Biotech Private Limited Hyderabad, then Vishal Biotech Nellore, She Biotech in Karnataka. These are some of the examples. Ozone Biotech in Faridabad, Sri Disha Biotech in Hyderabad, then Transcorport Technology Bangalore, Sun Biotechnology Limited in Delhi, then Gen Organic in Hyderabad. So there are large number. This is a name of only few important one. It is not just only that this is the limit. Many more companies are there that are producing these products on commercial level, on large scale. My dear participants, I want to emphasize here that in near future, the application of microbiology and biotechnology is great. The importance of these two disciplines will never be less. Always application would be more. So there is a much and great chance of availability of the microorganisms. There is a much more demand of the scientists, the microbiologists and biotechnologists. So this subject must be propagated in full form and full enthusiasm, so that the charm, the application should increase in the future also. These few words, I thank you a lot. So this is the first time that I am delivering a lecture in for the participants in Satna, and always I would have been there presenting my lecture, being physically present either in Bhopal, either in Satna, but it is only because of the effect of coronavirus. So because coronavirus is increasing day by day, so there is a great expectation of the people to all the microbiology that some new product, new thing will come in near future. Microbiologists can do, biotechnologists can do, and certainly I hope this will be done in near future. But always we have to be alert. And I wish a healthy life, a happy life. Thank you so much. Thank you a lot.
Thank you very much, sir, for your nice deliberation. And now we are moving towards the Q&A session. We will pick up 3-4 questions from the participants. So, sir, please resolve that. Arpit?
Yes, sir.
Yeah.
Thank you so much, sir. And yes, we are ready with the questions. The first question we have from participant. His name is Harsimranjit Singh. He is asking, sir, which biofertilizer is best for vegetable crops?
Which vegetable crop you want to select for the use by biofertilizer? There are certain restrictions for before using the biofertilizer. It must be agroclimatically suited. The biofertilizer produced in Tamil Nadu will not work in Haridwar. For application of biofertilizer, the microorganisms well adapted in the environment of Haridwar should be searched out and should be done. Please let me know which type of vegetable you want and where you want to apply on it. The nitrogen fixing biofertilizer, non-nitrogen fixing, free living, the fungal, the bacterial or viral.
Thank you, sir. So let the participant will decide and will move further. The next question we have, we are observing the climate change nowadays. So, sir, will climate change will become a threat for biopesticides and biofertilizers? in upcoming
Yes,
days?
yeah, certainly. Due to climate change, there is a rise in temperature. There is a rise in pollutants day by day in the soil. So, if we have produced biofertilizer once, it does not mean it will always work throughout the life. No, we have to search out time to time, because the efficiency of the biofertilizer, where the microorganisms have been used, it becomes a gradually decline with due course of time. Efficiency becomes a decline. So this work needs to be done continuously and examined in the field continuously.
और दूसरा उसका आगे प्रोजेक्ट आया डीएसटी से ये वो
Thank you so much, sir. Then the next question we have: what are the effect of groundwater pH? Do they have direct effect on the efficacy of biopesticides?
You know, the groundwater is declining day by day. It is going deeper and deeper. And when you are using the chemical fertilizers, chemical pesticide, and it percolates, it goes down, it gets mixed up with drinking water inside the earth. And if we are taking out the water, certainly there would be pesticide and chemicals. So for production of biofertilizer on commercial level, we have to decide which type of biopesticide or biofertilizer we have to use, in the form of liquid or in the form of solid. I told you in one of the pictures that the biofertilizers can be used in the form of granular powder, in the form of suspensions, in the form of foliar spray. So it depends upon the agriculture, the farmer, in what way and where they want to use. You want to apply on the foliar region, you want to apply in the soil. So water which you are using may or may not be contaminated by the heavy metals or any type of other dangerous chemicals. If dangerous chemicals or heavy metals are there, I suggest such water should not be used. Such water should not be used for preparation of biofertilizer, because that is other area of the research known as a bioremediation. In that condition, the microorganisms are used for remediation purpose, not for biofertilizer production purpose.
Thank you so much, sir. The next question we have: how much Indian farmers are well aware about biopesticides and biofertilizers nowadays?
Okay, the Indian farmers are aware neither for the pesticide usage, neither for biofertilizer application. They use the chemicals, biopesticide in free hand. That's why I told you the example of the Punjab. The recommendation is 77 kg per hectare, but they are using 177 kg per hectare, double amount. Even then, the production is not so good, because the soil has been totally degraded. No fertility is there, because soil has carrying capacity. The carrying capacity of the soil to carry the nutrients, to carry the organic material, to carry the clay minerals, it is degrading day by day. If soil has lost the carrying capacity, then the effect of chemical fertilizer would be high, and the application of biofertilizer or effect of biofertilizer would be low, because of these reasons. So before applying this biopesticide in the soil, we must know, we must understand the nature of the soil also, where we are going to apply, where we have to apply. So the farmers must be trained about the amount, about the agricultural situation, about the health of the soil, so that they can apply in recommended dose, not in free hand. Otherwise, production would be not up to the mark.
Thank you so much, sir. So that's all from the QA session. And over to you, Dr. Kamlesh.
Thank you, sir. Now I request Professor Anil, sir, for concluding remark on sir lecture.
It was a very nice lecture given by Professor R.C. Dubey on the biofertilizer and biopesticide, and he has also given the very good information regarding that the farmers, they are not aware about the how much amount of the biofertilizer or how much amount of the pesticides they have to use in the field, so that the problems of coming, like in Punjab, he has very well mentioned about the in Punjab that they are facing a lot of problem in Punjab for uh, of the biopesticides, and they are using much amount of the urea and other biofertilizer, means biofertilizer, not the biofertilizer. So this is very good information we have gathered from him, and I must give him thanks to for giving this such kind of information. Thank you very much, Professor Dubey. Thank you. Now our
Thank
next
you, Professor
speaker,
Anil
I
Prakash.
think our next speaker, Dr. Kamlesh, please introduce our next speaker. Thank you.
Yeah. Thank you, R.C. Dubey, sir. We are very much in depth. We have learned so many things from you, sir, and always you support us in many ways and also encourage us. You always come on our invitation and always inspire us in many ways. So thank you very much for being with us. And now we request our next speaker, Dr. Aradhana Mishra, ma'am. And before starting her lecture, I just would like to introduce in front of you. Dr. Aradhana Mishra is working as principal scientist in Division of Microbial Technology at CSIR Institute, that is National Botanical Research Institute in Lucknow. Dr. Aradhana Mishra holds a PhD in Bioscience in 2007 from Rani Durgavati University, Jabalpur. Dr. Mishra joined CSIR NBR as a senior scientist in January 2013 at Division of Microbial Technology. The research was focused on bio-inspired nanomaterial synthesis and plant microbe interaction during abiotic and biotic stresses. And she has been awarded for Women Scientist Fellowship Award by DST Government of India in 2007 and 11. Several major research projects have been completed by her. So far, Dr. Mishra has more than 51 publications in peer-reviewed SCI journals, and 7 book chapters and 4 patents have been granted. And she has been awarded several times for best research papers by CSIR. She is also editor in reputed journal like PLOS ONE and former editor in American Journal of Clinical Microbiology and Antimicrobials and Genetics and Molecular Biology Research. In November 2019, she was honored with prestigious Women Scientist Award 2020 by the Biotech Research Society India. Recently, she has been awarded with honored by Brahma Deva Samaj for her scientific contribution, uh, uh, and she has also developed 3 products on bioagent for Fusarium wilt disease control, herbal nano serum for skin care, herbal nano emulsions for controlling dermal pathogen. Her research area is basically related with plant microbe interactions based on Trichoderma fusent, and from this, her group is also involved in evaluation of efficacy of microbes for abiotic and biotic spaces. And Dr. Mishra worked in also green nanotechnology by using silver nanoparticles having bactericidal activity against MDR diseases, bacteria. Currently, Dr. Mishra has led product development by herbal lipid-based nano emulsion that has strong antimicrobial activity against phyto as well as dermal pathogens. So Dr. Mishra maintains cordial relationship between various parts of country, including CSIR, IITR, CSIR, welcome Aradhana ma'am to start her lecture. Please, ma'am.
Okay, we should share my slides here.
Yeah.
Can you hear me, Dr. Kamlesh?
Yeah, on right side you will see there is four
Yeah,
icon
I will.
from
I
above.
will. Okay, I'm going to share my slides.
Yeah, please. The slides were here, ma'am. Your screen was shared. You just closed it. Please again reshare.
Yes, I'm just trying to connect. There is
Okay, ma'am. Now it's fine. Now share your presentation.
now. Yeah,
Please minimize it and share your presentation.
yeah. I'm doing. Wait. It is okay now?
Yeah, your
Okay.
screen is here.
Okay. My slides are there.
No slides
My
are not.
first slide is
Just minimize that internet browser.
doing. I'm doing
Internet.
so. Wait. It is now okay. My
Not
slides are
yet.
there.
Not yet. It's coming. I think we have to wait.
Give me 1 minute. I'm doing. I minimized the screen that was earlier.
Ma'am, you have selected entire window option.
Yes, I have shared the slides. Share screen is here. Then I shared application window. Application window I have clicked. It is
You click entire window option. You just. Yeah. Now it's here.
it's okay?
Yeah. It's,
Okay.
yeah. Now it's.
Uh,
Thank you.
okay. First of all, Dr. Kamlesh, thank you very much for inviting me for sharing my talks with such esteemed personalities, Dr. Dubey, Anil Prakashar, and everybody who is listening to me. And it is very good morning to all of you. Very happy to listen R.C. Dubey, Dr. R.C. Dubey sir's lecture. And now I'm also focusing my talk on the intervention of biofertilizer and for the sustainable agriculture and food security. Basically, we are working on biocontrol agents, but this is the time for the upgradation. So we have introduced some nanotechnology for the biocontrol. And all the biocontrols are related on the microbes. And first of all, we talk about the relation about microbes, soil and fertilizer. Actually, soil is very good home for microbes. So different kind of microbes are there. Some are pathogenic, some are beneficial, and soil is affected by those and plants are also affected by those. So all microbes are not beneficial, all microbes are not pathogenic. But it is the mutual understanding between plant and soil that is going on naturally. But sometimes where the pathogenic bacteria attack to the plant, then problem started from there. And sometimes plant is not able to take the mineral from the soil, then problem is there. Then the microbe play the key role over there. It degrade the minerals and macronutrients from the soil and it provide to the plant. and plant provide root exudates to the microbes. This kind of mutual understanding is going on in the nature, and we also find some potent microbe for the treatment of the plant. Treatment for the where the pathogens damage the plant, and we have to introduce that microbe who they are able to control the microbes. But for the production of plant, it is general practice. Our farmers and our business, agro-business farmers, they all are using fertilizers. They are looking for the instant result and fast results, and they are using fertilizers. And fertilizer is giving the result, no doubt about it, but as well as the negative point of the fertilizer is continuous use of the fertilizer. It damages the soil as well as microbial microflora also. And gradually it disturbs the microbial community. If the microbial community is disturbed, and all the carbon chains and all the system that is through the food chain is automatically disturbed, and plant productivity, plant yield, soil quality, soil health, all those things are disturbed. So these three things are the key role of the nature, microbe, soil and fertilizer for the production of yield of the different of the crop. In general practices that I earlier discussed, that the numerous ways to improve the fertility of soil, they use volatile leaching, chemical fertilizer, cultivation, crop rotation, mulch. So those are the earlier practices that they are doing. And generally they introduce NPK for the better growth of the crop. And farmers are not thinking about the soil, and continuous excessive use of the fertilizer is going on. So what happened? Result in the Punjab and surrounding area, Haryana, somewhere in UP also, it is an alarming situation. Excessive use of the fertilizer is going to damage the soil, and finally it goes to the water bodies, and it goes to the food chain. Animal also take those water, and in plant it also present in the leaf, in the root and soil, and day by day it gradually consistency in the plant, and in that way it is continuously present in the plant, water, soil, and pH level is so high of the soil, and gradually it damages the soil finally. So this is the alarming situation in this area. The newspaper has covered excessive fertilizer use in poisoning Yamuna, fertilizer overuse eating with the Punjab soil health is degraded in the most region, soil health initiative wearing farmers of the fertilizers overuse, kind of. So being a scientist and based on the plant-based research institute, it is our responsibility to develop a product that should be beneficial for the plant, as well as it is eco-friendly and safe for the human also. So we have concentrated on the biocontrol project. So now we can understand this kind of chemical fertilizer that is present on my slides, urea, anhydrous ammonium, and superphosphate and diammonium phosphate. This kind of fertilizer finally goes to the water. When we apply urea in the water, it maintains in the waterlogging condition, and it produces the methane, CH4 gases. It degraded and it produces the methane. Denitrification process is there and produces methane, and lots of pollution is there, and methane has a very high potential for the damaging this air. So this kind of chemical fertilizer is in the practices, and we can see in this flow chart how this first it goes to the goes to the crop or the plant, then it gradually photodecomposition in the wind, and it goes to the water vapor, and it also goes to the water bodies, and in that way it is continuously involving in the food chain and polluting our water and animal system also. So we were discussing earlier how this urea is producing methane. In waterlogging condition, there is no oxygen. When we prepare nursery, and after that when the waterlogging condition in rice crop, we continuously maintain logging condition, and in that logging condition, methane gas is due to production and degradation of the urea, and final product of the nitrous oxide gases. So the nitrous oxide gas is the second potential element of creating the climate change pollution. So very alarming situation. So we have to be aware about using the chemical fertilizer, and we have to be aware about the we can stick to the biocontrol agents. So we think about the solution that the biofertilizer and that substance include microbes, which can help the plant growth promotions, and essential nutrients they can send to the plants, and living organisms, those who are mutual understanding with the plants, blue-green algae, this kind of things, they can be used for the biocontrol and biofertilizers. And as well as next step of our study is included to the herbal nanotechnology. Right, biocontrol is going on since last. But what is new about our study, that we have to also introduce nanotechnology, where everywhere we are depending on the nanotechnology and upgradation of the technology. So here we are also using nanotechnology. Few are researchers and scientists, they are using nanotechnology for reducing amount of the fertilizer. But we have shipped the fertilizer. We are looking for the substitute for the chemical fertilizer. So we are preparing herbal lipid and synthesizing nanoemulsions. That the nanoemulsion is able to control the phytopathogen and crop production, better yield. Actually, biofertilizer works through the roots. When biofertilizer is in the root, it applied to the seed and the surface or the soil, it colonizes the root. In this picture, we can see the bacteria colonize to the root, and it uses the root exudates and provide all minerals and water to the plant. In that way, like phosphorus and other element that is plant is not going to take directly, with the help of this bacteria will degrade the element and it will provide to the plant. So in that way is very easy for the plant. And of course, biofertilizer is an alternative way, and it is the substitute of the chemical fertilizer. And crop yield has been reported for the 50% better than if we are using only biofertilizers. And after using 3-4 years continuous of the biofertilizers inoculum, sufficient growth and multiplication is automatically remain in the soil, and soil become so fertile and healthy. pH will also maintain because there is no harsh chemical is inside the soil. Mainly we are generally researchers and already reported our and phosphate-solubilizing bacteria, Trichoderma, Pseudomonas, Bacillus. Those potential bacteria has been already reported for the biocontrol preparations and introducing to the field for the plant growth promotion and pathogen control. Major difference between chemical fertilizer and organic or the biofertilizer is the fertilizer reaches to the water bodies and contaminate all my pond and other water bodies. It goes to the animal, and animal through the milk it comes to the finally to the human. And here in biocontrol there is no risk. There is no risk with chemical fertilizer. So we are safe when we are using biofertilizer. In our lab, we self-prepare the biofertilizer. We have taken patent for this trichoderma for the of nutrition value in the chickpea, and this trichoderma has been grows in the plate, and after the preparation of autoclaving of the bajra, we mix with this and prepare the mixture, and after preparing the mixture, we can prepare in the seed coating for seed and for the soil application, for the foliar application, and we can also use in the water also. So it is our self-prepared biofertilizer that we are using, and we provide to the farmers during Kisan Mela and other. Biofertilizers are used to the seed treatments, root treatments, and soil treatments. In soil, we can use cocopeat, natural cow dung, water, and those kind of carrier we can use for the application of this biofertilizer. And seed coating, we can mix with seed this formulation for the 30 minutes and after that before sowing. And if we are preparing a nursery for something, so you put out the nursery outside of the before plantation, dip that small nursery in the tub, and water dissolving this formulation, we can treat like this. This is our product that we have prepared. Have used the Pseudomonas, and this is the liquid that we can use to the foliar spray and the root also, in the soil also. So this is the product of our that we have prepared, and we have got patent for this also. And this is the herbal nanoemulsion. It is the herbal nanoemulsion that we are using to cure the different kind of phytopathogen. Presently, we have tested on the Alternaria brassicicola, Alternaria solani, and this nanoemulsion was sufficient. We have used the peppermint oil and prepared the nanoemulsion. This is our team that we, and it was my introduction, but now I'm going to present the work that we have published and got good results. And this is our team that we have included. Some students are working on biocontrol, and some students are working on nanotechnology. So this is the endophyte mediated modulation that we have introduced the bacteria, and we have got very good results in Vidania somnifera in the presence of consortium of the bacteria. And we can see here, this is the control, and Alternaria alternata was the infection has been given to this plant. Here the simple control, there is no infection, healthy plant. Here we have used the bacteria, and here we have used with pathogen and bacteria. Consortia of bacteria, 2 bacteria, and here we have used alone 1 bacteria. Here with pathogen, and here alone bacteria, and here with pathogen separate bacteria. So 2 kind of set we have taken for the study. And the Pseudomonas group and Bacillus we have taken. Here the only Pseudomonas and bacteria we are evaluating that the consortia of the bacteria is going to give any positive or negative. So we have found that the consortia of the bacteria were able to give the positive effect to the plant. When we infect this plant, after that we have alternata, we have infected the plant. Alternaria alternata infection has been given and applied the both bacteria, Pseudomonas and Bacillus, and we found this consortia was able to control the pathogen. And individual those bacteria were also showing very good and cooperative result to the plant. And this work was published in Applied and Environmental Microbiology. And this is the microscopic study to find out the live and dead cell study. And in the different way, we have got and confirmed that the consortia of the Pseudomonas and Alternaria alternata has been also infected, and Pseudomonas and Bacillus both were separately able to control the disease. But when we apply Bacillus and Pseudomonas, then infection was completely able to control. So the consortia of the Pseudomonas and Bacillus were able to control the Alternaria alternata in the Vidania somnifera. And this is the again confirmation of the plant. Here we can see the mycelia of the Alternaria alternata. It is the healthy mycelia. When we are introducing the Bacillus aminoliquefacius, then it getting start punctured. This mycelia is punctured here, and bacteria is here over. Bacteria is covering the mycelia, and it is mycelia of the pathogen has been punctured and gradually damaged. When we introduce Pseudomonas fluorescens, then we see the mycelia has been completely damaged and squeezed, and it is completely damaged. So we can confirm through the some studies that the Pseudomonas fluorescensis and Bacillus aminoliquefacius both were able to control and inhibit the Alternaria alternata. Again, we studies where when we are applying those microbial consortia, the microbes to the plant, where they were able to produce, where they were able to provide the nutrient also. So yes, though two varieties were taken for this, Hina and Kiran. Kiran was these are the two rice varieties, and Kiran was the sensitive variety. Hina was the resident variety. First, 100% nutrition were provided alone rice. 100% nutrition was provided with biofertilizer. So in this picture, we can also differentiate where the biofertilizer trichoderma has been given. The rice plant was much healthy. In sensitive case also, trichoderma of BF biofertilizer was playing better role and in the rice plant. Here we have reduced 50% nutrition only. Here also 50% nutrition, but we have introduced biofertilizer. So this biofertilizer was able to cope up the plant for the healthiest. And sensitive case also. Here there is no nutrition. In control condition, there was no rice growth is there. But here when we are introducing biofertilizer without nutrition, the 70% growth was success in the resistant Hina variety. In the sensitive case also, the trichoderma was able to cope up the plant, and 0% of the nutrition condition, the rice were comparatively less, but 20% growth was observed. So we have also confirmed this study that we have confirmed this study for our introducing trichoderma in for the abiotic stress. It was also it was again published another work in Plant Molecular Biology Reporter. It was the matter of abiotic stress. We have taken stress as the high CO2 condition and introduced trichoderma, and we have phase facility, free air carbon enrichment system. We maintain 550 ppm carbon dioxide and grow the rice over there. This is the section of the rice root. So we can clearly see where we are not using trichoderma, when we are not helping any biofertilizer in normal condition. Here we can see pith region is damaged and the cell is damaged. So this is the disintegrated kind of. Here complete integrated section we can see strengthen. All the number of metaxylon are also increased here. So here strengthened plant and healthy plant. Here the rough surface is there. So cortical and in cortical region we can see the difference. Here it is damaged, here it is strengthened. In that way we can also confirm through the same study of the root in high CO2 condition that trichoderma biofertilizer was able to support the plant. As earlier slides, you don't confuse. It is the different kind of work. It is the nutrition stress, and we published is work in plant physiology and biochemistry, and with my team. The first author was my student. Next, high CO2 condition. Again, we introduced the biofertilizer trichoderma, and it was the result was amazing. And this is the picture of the Hina and Kiran that some already I have shown in the slides. Ambient condition and ambient condition with trichoderma, elevated in the sense high CO2 condition, and high CO2 condition with trichoderma, and in the Kiran the same way. So we can see here the growth is much better. Then we move to the tomato plant, and concern was that the tomato wilt to be controlled, where it was caused by the fusarium. So we introduced here bacteria, and we continuously studied and find that the bacteria Bacillus was able to control this fusarium, and the study was confirmed through the first microscopic study. This is the control, and this is introducing with bacteria, and it is the study with the pathogen. When pathogen alone is there, plant was damaged, but when here we can see pith region is damaged. But here when we are introducing bacteria, the microscopic study reveals that the complete strengthen has been plant has got. And this is the treatment of the chemical fertilizer. We have compared with the biofertilizer and chemical fertilizer. So plant was much better condition in the biological control system. Then now we have started that the nanotechnology work also. So this work was published in the plant physiology and biochemistry, and Madhuri was the first author and used silver nanoparticle, but it was synthesized through the trichoderma, because that nanoparticle was in various amount, but metabolite of the trichoderma was coated that nanoparticle, and we treated it. So early blight disease was treated with this metabolite coating nanoparticle. Here we can see the control. Again, here we can see the control, and here the plant, here the plant pathogen only, here the silver nanoparticle alone, here pathogen plus silver nanoparticle, here pathogen alone. So it is badly affecting the plant, but when we are treating with silver nanoparticle with trichodermal metabolite, it is healthy and coping up. The zoom image of this plant, it is infected and it is treated with silver nanoparticle. The leaf detach assay has been also done. Healthy plant, only silver nanoparticle, and pathogen infected but treated with silver nanoparticle. And this is no treatment and pathogen. And the same way the experiment was going on, and we shifted to the herbal lipid, and we prepared nanoemulsion. Here we have synthesized and prepared the nanoemulsions. This is the different lipid, and this is the peppermint oil lipid. So two kind of nanoemulsion we prepared and confirmed through the 10 studies, when we found that the 50 nanometer size was found, and we continue our study. Here we have used the early blight disease management by the herbal nanoemulsion in tomato, and this is the control. And in control we can see Alternaria solani was well growing, and when we are using 0.25% of the nanoemulsion, it is able to grow but not quite well. But when we are using 0.5%, it is lesser than earlier. When we are using 0.75%, it is not growing. When we are using 1%, it is not able to grow. This is the same day picture, same day inoculation. But in this plate, we can easily see when we are giving nanoemulsion treatment to the Alternaria solani, it is reduced. So the 0.5 to 2.5% nanoemulsion were significant to inhibit the growth of the infection of Alternaria solani. And this in leaf infection, we can also see the 3rd day result, and this is the 5th day result. In control, we are giving infection, there is no improvement. When we are giving the nanoemulsion treatment, so at 1% it is healthy and controlled also. But when we are exceeding the concentration of the nanoemulsion, leaf also got damaged. So we have to be alert and aware to standardize and optimize what concentration is required to treat the plant. So the 1% of the peppermint oil nanoemulsion dose was sufficient to inhibit the fungal growth. And this work was recently published in Industrial Crop and Products with my student Shipra Pandey. And this is the next picture of this test that we have experimented plotted in earlier slides. Here we can see we have taken the leaf in control. We are trying to find out the pathogen CFU, if there is pathogen in the leaf or not. Control, when we are not giving any infection, there is no. Carbendazim is the chemical fertilizer that we have given to the plant alone, no infection. There is no phytopathogen CFU. Alone peppermint oil nanoemulsion, there is no infection chances. But in the leaf that we have given the Alternaria solani infection, we can see the growth is higher, and spores of the Alternaria solani was found because in this leaf already given the infection. And here, infection with treatment of chemical fungicide, carbendazim, less amount. But when we are using Alternaria solani with peppermint nanoemulsion, very less amount, and it is completely inhibited by this nanoemulsion. So on the leaf, there was no minimum spores of the Alternaria solani was found. In this plant test, we can also see. Control plant treating with alone, there is no infection here, of peppermint nanoemulsion, carbendazim, it is the chemical fungicide, it is the infection, Alternaria solani, Alternaria solani treated with the chemical fungicide, carbendazim, and here Alternaria solani treating with nanoemulsion, peppermint nanoemulsion, and we can see the result. These are the newspapers that covered the recent study that we are preparing. Green nanoparticle applying trichoderma is able to control the fungi, and this kind of thing. This is our visit, and we put the biofertilizers to the Kisans and Kisan Mela workshops we provide, and we visit the field, interact with farmers, how the treatment is going on, what is the effect. So some people are very excited, some farmers are very excited, treatment was very good. Some educated farmers, rich farmers are also very much happy with these treatments, and we report in different kind of meetings. This kind of work that I shared with all of you, and visit in MP, UP, in different places. So I request you stop chemical fertilizer use and aware people and farmers for the biofertilizers. Thank you. Any question?
Thank you very much, ma'am, for this very important lecture. And we have learned so many you can say that practical approaches regarding how can we conduct our research studies related with biofertilizer, and yes, of course, related with nano biofertilizers. So, Arpit, may I request you to pick up 2-3 questions so that ma'am can give answers regarding from the participants.
Sure, sir. Thank you so much, ma'am. And the questions we have from the participants, the first question we have, how the shelf life and delivery of biofertilizers has been improved by nanotechnology?
We have applied the biofertilizer separately and nanotechnology separately. But if you are asking those things, we can put our nanotized material, nanomaterials like curcumin or the silver nanoparticles, introducing of those kind of things improve the shelf life of the biofertilizer. No doubt about it. Are you able to hear me?
Ma'am, your voice was actually, ma'am, your voice was not clear and audible. So if you can please repeat that. Aradhana, ma'am, your voice is not audible.
Arpit?
Yes, ma'am. Ma'am, yes, ma'am. Now you are audible, ma'am. So if you can just repeat the answer, because at that time your voice was getting too much. Yeah. Thank you.
Yeah, sure. So, as per my research experience, and we can use curcumin nanoparticle, nanomaterial, and silver nanoparticle in trace amount that is safe concentration. It may enhance the shelf life of the biofertilizer. Still, we are working on, and different kind of polymer you can also polymer nanotizing, and you can add to your fertilizer. Those kind of things improve the shelf life of the biofertilizer.
Thank you so much, ma'am. The next question we have: how to study the fate of biofertilizers, as in the behavior in soil and environment?
Can you repeat your question? Play something you asked that I was not able to understand.
Ma'am, how to study the fate and behavior of biofertilizer in soil as well as in environment?
Yeah, first you go for pot trial. After that, soil. You put the field trial, and not only micro plot, in big plot in agriculture university. That is the authentic way to evaluate your biofertilizer, as we are doing. We are putting our field trial in agriculture universities and taking the disease prone plant, what disease you are studying. Don't give artificial infection. Are you able to hear me?
Yes, ma'am. So thank you so much, ma'am. And that's all from the QA session for this. And over to you, Dr. Kamlesh Chaudhary, sir.
Yeah, definitely. Thank you very much, ma'am, for this inspirational and insightful talk. Now I'm handing over the mic to our convener, Professor Ranit Prakash, sir, for his concluding remark.
Thank you very much, Kamlesh. One question I would like to ask Aradhana ji. One is,
yes, sir, sure.
You have used the silver nanoparticle. That is,
yes, sir.
You have made these silver nanoparticles of your culture or the bioactive compound of your culture. What?
So we have prepared synthesized the nanoparticle with the help of trichoderma metabolite, and we have tested and published the paper about the safe so in soil microflora also biolog study. So we have applied in the soil.
Okay, but you have used the
Yeah, we are
metabolite
using
and the trichoderma.
yes sir, yes sir.
Okay, okay. Thank you very much, and we have really a very good and inspired lecture you have given.
Thank you
And
so much, sir.
related to these nanoparticles and biofertilizers, because you have used it is very well known. Trichoderma is very well known to use as a
Yes sir.
to inhibit the growth of the pathogens and others, and it is very well known. But you have used the nano fertilizer, the nano bioparticles of that to inhibit the growth of the phytopathogen. So it is a really very good and nice work you have done it, and I saw
Thank
your
you, sir.
team also. It is a very good team you have developed it, and with these words, I must congratulate you for giving such a nice lecture. So now I will again hand over the mic to the Kamlesh for introducing our next speaker. Thank you very much. And Dr.
Thank you
Aradhana,
very
you
much.
will just your email ID is required.
I will send you, sir.
Okay, thank you.
Yeah. Thank you very much, sir, for this concluding remark, and thank you very much once again to Dr. Aradhana Mishra ma'am for giving your important time and for your nice deliberation with our attendees. And I hope that we have learned so many things from you. And in the future, we will again contact you for detailed lectures so that we would like to learn more applications from you. So
Sure.
now now I'm welcoming our next speaker, Dr. Mahavir Sharma sir, who is joined us since morning. And before starting his lecture, I would like to introduce Mahavir sir. Dr. Mahavir Pratap Sharma is currently working in Agriculture Research Service as a principal scientist in agricultural microbiology at Indian Institute of Soybean Research Indore, that is an ICAR institution, and which is comes under Ministry of Agriculture and Farmers Welfare, Government of India. Dr. Sharma earned his undergraduate and master's degree in agriculture from Rajasthan Agricultural University, Bikaner, and his PhD from Gwalior. He has received gold medal award during his master's program. Dr. Sharma has started his career in mycorrhizal research at University of Delhi and TERI, New Delhi, and after serving TERI for 11 years, he moved to ICR and joined in ARS as senior microbiologist. He has specialized in soil microbiological research involving the use of plant growth promoting microbes, particularly arbuscular mycorrhizal fungi, in improved plant growth, soil carbon sequestration, drought tolerance, and overall productivity of crops. He has been awarded many awards like Best Paper Presentation, Travel Grant Awards for visiting abroad in various scientific meetings, conferences, and has won several external research grants from DBT, DST, ICR for his research on applied aspects of plant arbuscular mycorrhizal fungi, microbe interaction. He has also been awarded the prestigious DBT Crest Award in 2013 by Department of Biotechnology, Government of India, to work abroad on signature fatty acid biomarkers to assess live biomass of AMF and soil microbial community changes in log-term formal system trials. There are about 90 important research articles published in referred journals, magazines, and reviews articles in books of international repute, and has many microbial accessions with NCBI database and cultures deposited in international microbial repository authorities to his credit. So now I welcoming him to give his expertise and present his expertise, and we will honored by learning his particular talk. We welcome you, sir. Now over to you.
Thank you. I am audible to you all.
Yes, sir.
Okay.
share to entire screen.
Yeah, select entire screen, then start presentation. Minimize this window, sir. No, that was fine actually.
Okay, okay.
Select entire screen, then minimize internet browser and start your presentation, sir. Minimize this window, sir.
Minimize this window. I have.
Just seconds. Please minimize it. Now open your presentation. It will be here. Yes, sir. Full screen.
दिख रहा
Please,
है?
yes, sir. Please hide this bar, sir, which is coming over slide. Click on hide. No, that window. Yeah.
Okay.
Now full screen mode on. Yes, sir. Now starts.
So first of all, very good afternoon to all of you, and many thanks to the organizers to providing me this opportunity to share my views on one of the biofertilizer, which is mycorrhiza. The morning two talks was really very introductory as well as very inspiring in the sense my talk is entirely different than the other two. So hope I will justify my time to speak about this new biofertilizer. So mycorrhiza, is a symbiotic action with higher plants, and it helps. First, I would like to tell you the structure of my presentation, the challenges and importance of mycorrhizal fungi. Then I will discuss about the current taxonomic position. Then how to manage native and resident mycorrhizal strains. As in the morning, Professor Dubey has emphasized the use of native strains, the regional specific strains. So as we all know, if a strain belongs to exotic origin, it may not be survive in our conditions. So the well-adapted strains has to be considered for using biopesticide or biofertilizer. Then I will discuss how mycorrhiza is useful in climate mitigation, the carbon mitigation, where I will emphasize the use of glomalin, and we have also one case study where we have assessed the importance of glomalin in carbon sequestration. Then I will also touch upon how to assess the PLFA microbial communities, and of course, if you have a product, how to develop a product and how to test the quality of its, the inocula and the future direction of research. So as we know, Dr. Aradhana, she has already highlighted, and other two, Dr. Dubey also, the intensive use of external inputs, resources, pesticides, irrigation mechanization. This all part of green revolution has not only sustained our crop production, but at the same time deteriorated our soil environment. So major challenge is we have biofertilizers, we have biopesticides. Only thing, we have to increase the input use efficiency. That is the only way out. And for that, better delivery system has to be there. Like Dr. Aradhana, she has already explained one of the formulation, the use of nano synthesized particles that will not only enhance the efficiency, but also improve the shelf life.
So as we know that there will be demand for food and the stagnated resource base. At present, we have 142 million hectare area under agriculture, and it will further reduce because the anthropogenic activities as well as urbanization is taking place, so it will further reduce. So there is the only possibility to increase the input use efficiency. There is the role we have to play a great deal. So as a scientist, we have to work how to increase the input use efficiency with better microbes, well adapted microbes, and so that the microbes and the farmers both can work together. And the microbes which are present in the soil, fungal or bacteria, they are invisible. Only visible is the impact, that is the yield. And if you compare the macroorganisms like insect, for example, BT cotton. So we can see our naked eyes the insect and the impact of the BT. But in this case, it is very difficult. Quorum sensing and other things are, the scientific, the way out to understand the mechanism. It is there. But for farmers' point of view, only the yield is an indicator. So we have to be very careful while delivering our product. Third-party evaluation is must. As one of the experiment, Dr. Aradhana, she has explained, under the phase conditions, the use of microbes at elevated CO2 up to 600 ppm has a tremendous role. So that is must, and we have to look how to reduce the terrestrial soil carbon as a carbon sequester, where I will be touching upon how mycorrhiza can be used as a carbon mitigator, but at the same time also sustain the crop productivity. As we see at present, we have about 413 ppm CO2 in the atmosphere, and this will further increase. And the plants, for example, C3 plant, they will behave like a C4 plant when CO2 will increase beyond 400 ppm. So soil quality, and we need to maintain its biological soil health. Organic farming is one of the alternative. As per the Kyoto Protocol, we have to mitigate the CO2 as a part of UNFCCC protocol. We are also, or Indian, as Indian, we are also the part of this protocol. So we need to have a technology, for example, how to increase the carbon sequestration through microbes without compromising the crop productivity. So there, the role of these microbes play an important role. The reclamation, remediation, or bioremediation is also one of the areas where we as a microbiologist can also do a greater role. So I will emphasize in my talk how mycorrhiza is highly relevant, not only for crop production, but also for carbon mitigation. As we know, this is one of the fungal biofertilizer, has been notified by Government of India during 2010, and since then it is being supplied in the market by the commercial companies. At the same time, many things are happening. The quality is becoming very poor, almost all biofertilizers, and there is no mechanism where referral labs are there. Only few labs are there. The regulatory mechanism in our country is very strong, but the monitoring is very poor. very difficult to say at this point. As a microbiologist, we always blame that microbes doesn't work. But the only thing that spurious microbes are being supplied in the market. So in case of mycorrhiza, as we know, more than 80% plants are associated with this fungi. Is obligate combined. It requires host for its own survival. At the same time, it enhances the plant growth as well as mobilize the nutrients, particularly phosphorus, which is immobile form. So mycorrhiza is not a P solubilizer, it is a P mobilizer. So P solubilizer is the bacteria which can travel through the mycorrhizal hyphal network and then reach to the plant. So it is acting as a vehicle for the plant and through the hyphal extended root system. When plant is there, mycorrhiza is there, so both will talk and then they develop a symbiosis. And only mycorrhiza needs carbon in terms of root exudates, which is supplied by the plant. And in turn, this fungi through the extended root system add to the plant root system and it will better cope up with the stress by supplying the water as well as the immobile nutrients. For example, phosphorus is the major one, and zinc, nitrogen, and other micronutrients are also enhances through this mycorrhization. It also improves the plant physiological status of the plant by improving the cytokinins, changes in soil solute or the plant physiological changes. For example, it increases the proline and all that impart the tolerance to the plant. It also helps to provide the tolerance against the root diseases, and many reviews has been published on this. Apart from this, it produces a humic compound which we call organic glue, which is a glomalin that helps in binding the soil particles or helps in the soil aggregation. And this is how mycorrhiza is useful as a part of carbon sequester, and which is a new area, although the work started after 2000, and at present in India, 2 laboratories are working, but it has a great potential. As I told you that it requires the carbon which is available from the plant, and in turn supplied the nutrient to the growing plant. So in the sense, when the conditions is good for the plant, it is also win-win situation for the fungi. So my point here, want to highlight, when the phosphorus is very high, the plant will grow, but at the same time, the fungal may not be happy because there is a negative correlation. So, and also plant doesn't require the phosphorus, so it is just going waste in the soil. But indirectly, in our soils are P deficient. So whatever phosphorus is there, it get immobilized, fixed form. So 70% goes waste. So this mycorrhiza helps mobilize this phosphorus due to the plant. And a lot of work has been carried out. If you see the example of mycorrhizal research all over the world, the maximum research has been carried out on corn, cotton, soybean, and the phosphorus. After that, a lot of work has been published. And for example, in disease aspect, soil carbon sequestration, drought, because it is also acting as a synergism with other bacterial cultures. So both can be used together as a microbial consortia. If you see the colonized root section of any plant, when the plant is there, mycorrhiza spore is there, first it will germinate and form a entry which we call aprisorium, and after that it becomes inter and intracellular and makes the structure. The major structure is arbuscules. That's why we call arbuscular mycorrhiza. Many people still telling where vesicular arbuscular mycorrhiza, which is not correct at present, because all mycorrhizal fungi, they produce arbuscules, but not the vesicles. So therefore, the correct term is AMF. Vesicles are also very important. These are the terminal swelling of hyphal structures. So these are storage organs, but where vesicles are not there, the arbuscules are performing both the jobs. They are exchanging the nutrients as well as storing. Where we do find mycorrhizal fungi, these are present everywhere. I told you more than 80% plant families do form mycorrhiza. Some exceptions are there, like Chenopodiaceae family, Cruciferaceae family. Mycorrhiza is less than 10%, means the colonization. So those are non-host plants. So better we should avoid in the cropping system. For example, the mustard. Mustard is a good oilseed crop, but if you grow always mustard every rotation, then the mycorrhizal population will decline drastically. So my point is here to highlight that the use of sustained cropping system is very much important, which is not only helping the mycorrhiza, but also other bacterial communities. In that sense, we have studied in the long-term field trials also, and we have observed that the use of mycorrhizal host is very much important in sustaining the crop productivity. As far as the current taxonomic position is concerned, at present we have 30 genera. About 200 species has been characterized based on morphological and molecular features. Out of 31 genera, we have 8 genera. Out of 31, 8 from glomerales and 10 from gigasporales and 10 from glomus. So this is a new latest article published in Mycotoxin 2012. After that, one publication also appeared, but it has become also a game changer for taxonomy purpose. I am telling you why. There are many groups are there. The Chris Watt group, he is defining different types of AMF species. So they have their own names. For example, Glomus introduces, now it is being used, renamed as a Rhizophagus irregularis. So there are a lot of confusion is there. Although I am not very much expert in the taxonomy, but this is the one area which is lacking in our country. Earlier, Professor K.G. Mukherjee, with whom I worked, started my career. He was the man who used to work on taxonomy and published many species on his name, mainly on morphological basis. But now molecular is more important. Now, how to manage the native AMF? One experience we got from long-term field trial on soybean, and in this experiment, we have observed that when we use the maize in the crop rotation, the glomus was found to be more predominant. So whenever we want to recommend the biofertilizer, first we have to use the adapted strain. In this case, we optimize the production system as well as characterize the species from the long-term field trials. So this glomus introduces is becoming most predominant, which is a Rhizophagus irregularis, and can be used for mass production program. This was the long-term trial, soybean maize cropping system, and this is being practiced at our place more than 15 years. My point here, after this study, we have observed that the glomus introduces or Rhizophagus irregularis can be used as a potential AMF helping to improve the crop productivity in the soybean based cropping system. So this can be targeted in production program. This we published long back, and a lot of citations are there from this paper. Here I would like to highlight the soybean yield versus the inoculum potential. Inoculum potential is the vigor of or the power of mycorrhiza to form infective propagules. That is the quality parameter, because when the spores are there, if they are not germinating, it has no use. So the inoculum potential is very much important, and that is the main quality parameter, or indirectly you can call the MPN method, which is being used globally. So here we observed that when we reduce the tillage system or tillage practices and include maize in the rotation, the mycorrhizal biomass or the inoculum potential increases significantly. So after that, we observed there must be some mycorrhiza must be predominant from soybean and maize. Then we have enriched the population by growing the plant separately, and then we assessed what type of predominant AMF are there. We grow the plant in the potting mixer, and then we assessed the AMF, the predominance. This is the universal protocol we use using the trap plant. In this case, soybean maize was used because our objective was different, to know which mycorrhiza is predominant so that we can target for mass production. This is a method, the microscopic method, using the stereozoom, and then we can characterize the spores and then use further. You see, out of all species we observed, in maize all 4 was found to be predominant. The Phenoliformis mozi, earlier name of Glomus mozi, Glomus geosporum, and Rhizophagus irregularis, and Glomus species. And species level we could not recognize. But in case of soybean, the same Rhizophagus irregularis was found to be predominant. So it means that this species we can use in this crop for this cropping system. So this is how we have characterized, and then we have target this AMF species for mass production using the pot culture method. The crop rotation influences the mycorrhizal parameters, like maize showed higher AMF biomass in terms of colonization, infectivity potential, which also coincides with higher grain yield. The conventional reduced tillage found to be better in terms of maintaining higher AMF biomass than conventional tillage, which reduces AMF biomass. When you till the soil frequently, the hyphal biomass get disturbed. So it is advisable to minimize the tillage practices so that you can harness the full potential of these mycorrhizal fungi. The Glomus irregularis or Rhizophagus irregularis found to be a dominant species colonizing both soybean and maize, and hence we used for inoculation in soybean based cropping system. This is a consortia of 2 species, mainly the Glomus introduces and the Glomus phenoliformis, or the phenoliformis mozi. Phenoliformis mozi. I think through this system, I am feeling humming sound. It seems that I am repeating the same thing again. Anyway, so the next is there are some prerequisites how to apply the mycorrhiza. Many of us may not be aware that the colonization is a poor indicator for growth. If you see the papers being published in Indian journals, if the colonization is higher, the growth is also higher. It doesn't happen always. So it is a poor indicator. It is a good indicator when you are using a host for mass production purpose. But for the growth response purpose, it may not hold true. AMF are site-specific than host. As I told you earlier, the adapted strains are not only based on host, but also mainly for site-specific. It means the soil contains more heavy metals. There must be AMF which are tolerant to those metals. So therefore, the site-specific AMF has to be considered. Mycorrhiza has more potential in transplanted crops grown in marginal soils, in the sense when the phosphorus is high, the mycorrhiza will not perform. So the marginal soils are poor in nutrients. Mycorrhiza has a poor and proven application in plant production and micro-propagated industry. It has been published widely. It can save the biological hardening time. It can save fertilizer saving, also provide drought tolerance. It helps in bioremediation and carbon sequestration. The correct methodology and dosage optimization and assess the vigor of AMF inocula needs to be very much important and need to a highly reproducible technique. We are lacking in this area, because if the spores are there, if those are hollow and they are not colonizer, so no use. So the correct protocol has to be must. Superior mycorrhizal species can be screened based on phosphate uptake than the colonizing ability. So this we have published also, and it has been widely recognized by other groups also. So when we screen the AMF for growth purpose, the phosphorus uptake is must. So you can assess the plant responsiveness based on phosphorus availability. These are some of the points to remember. The correct protocols for practicing AMF is must. Nutrient profile and host site specific preference has to be there. Timing before seed germination, pathogen introduction is must. When you use mycorrhiza after the transplanting or after the seed sowing, it will go waste. So there is no point. So it has to be applied just below the seed. Inoculum components, as we know, the mycorrhiza, the spores, hyphae, infected root segment. These are the major components of any AMF inoculum. So therefore, you cannot say that spores are the only inoculum. So these are the part of an inoculum component. Those has to be optimized before applying for any crop. Inoculum placement or method of application is very much important. Has to be applied just below the seed, or in case of transplanted crops, just below the root. This is how the seed treatment you can apply the mycorrhiza as a seed encapsulation. You can also use mycorrhiza in rice as a bare root dip treatment. You can make a solution of mycorrhiza and dip the seedling and then transplant it. AMF evaluation for plant. As I said you that mycorrhizal dependency, which is highly variable, it is influenced by several factors like plant species, soil fertility, and fungal strain. Therefore, if the phosphorus is very high, the plant is less dependent for growth through mycorrhizal fungi. So we have to be very careful while using the mycorrhiza for the growth enhancement purpose. Root geometry is also very much important, because these mycorrhizal fungi, they add to the plant roots and extended root. So therefore, some plants are highly dependent on mycorrhiza, some are less. So the root system is very much important. So we need to optimize mycorrhizal association for selecting a highly microtrophic plant or cultivars suitable for breeding material for releasing improved varieties. Ability to assimilate phosphate under low input condition. In our country, the breeding varieties are being evolved under the high fertility conditions. So
sometimes the variety may not perform for the mycorrhizal purpose. Mycorrhizal purpose in the sense, I am telling that when any variety is breeding under high fertilizer input condition, so that variety is not taking the benefit of mycorrhiza. So if you are having 10 AVT2 entries, advanced varietal lines, and out of 10, some may be good, but that you are giving the chance while using the high fertility levels. So the marginal fertility level is must for breeding purpose, so that you can take the advantage of mycorrhiza. We can apply the mycorrhiza in the application of mycorrhiza is well documented. Mycorrhizal inoculum in the forestry plant is well known, as we know that in the container grown seedling, the mycorrhiza can perform very well. When you transplant under field condition, the plants are already colonized, so you can really getting the benefit. But in case of cereal crops, it is becoming a challenge for all of us, because the inoculum requirement is much, and also the condition of the soil in terms of the fertility level is varied. So that is very critical. We also worked on use of mycorrhiza in micropropagated strawberry, and we have got the tremendous potential of this mycorrhiza by saving the fertilizer inputs. In case of onion, 25%, and we have published also way back. And in case of acacia, which is a forestry plant, you see here there is a saving of 20%, almost 50% phosphatic fertilizer. You see the response at the native P level is higher than the higher levels of P. So here I would like to highlight that the phosphorus level is very critical for taking the benefit of mycorrhiza, and mycorrhiza can substantiate up to 50% of phosphatic fertilizers when you use the potential mycorrhiza. As I told you that micropropagated industry, it can help in the survival of the plant during the hardening period. Because what happens when we take out the plant from rooting media and then after hardening we transplant in the field, so the most of the roots are lateral roots, and taproot comes later, and by the time plant not survived and collapse. So this mycorrhiza, if colonized during the hardening period, it helps to bring down the taproot faster, and the plant can withstand under the stress conditions. For example, low moisture and other biotic stresses. In case of strawberry, which we have published long back, that 50 kg phosphatic per hectare we applied, that was inadequate for strawberry. So it depends on the crop to crop. When we used 150 kg phosphorus per hectare, the response was same when you use mycorrhiza at 70% phosphorus per hectare. So almost 30 to 35% cost economic when you use mycorrhiza. And this is a very good experiment. I would like to highlight that if you want to evaluate the response of mycorrhiza, input versus output response, using the non-linear regression analysis, very good tool to get the benefit of mycorrhiza. If you have a particular agroclimatic condition and a non-phosphorus level and use mycorrhiza, you can optimize for all the plant species what is the internal requirement, what is the external requirement, and the saving of phosphorus. At the native level, the plant get differently benefited through mycorrhiza as compared to the higher levels of phosphorus. So therefore, the mixtureless response, if you follow for any field experimentation, you will get good response or a clear-cut picture of the mycorrhizal application response. This was the experiment of strawberry, and then we observed 30 to 35%, and we can save the phosphatic fertilizers. Mycorrhiza has also benefit to for the control of nematodes. This is one plant, Polonia, which is a very good agroforestry plant. Till date, we do not have any solution because it is highly infected with nematodes. Yeah, but if we use a mycorrhiza well colonized plant, then it can help to withstand under this nematode infestation. It can also be used as a very well with the Pasturia penetrans. The reports are there. The mycorrhiza along with Pasturia penetrans can also reduce the root knot nematode infestation. Now the next portion of my talk is how the mycorrhiza is relevant in carbon sequestration. As I told you, the mycorrhiza which produce a recalcitrant protein we call glomalin, which is a glycoprotein that enhances soil aggregation and sequesters soil carbon. This glomalin act as a glue and join soil aggregates together. If you see 100 years back, we were not aware how the sand dunes are being stabilized, but that time we were not aware that it was because of this glue, and that was playing a greater role. And this glomalin can, highly resistant to decomposition. It can survive to 42 years in the soil, and this represents up to 30% of total soil organic carbon. So you can imagine the potential of this mycorrhiza glomalin in the soil organic biomass carbon. This glomalin, you see, the mainly present in the hyphal biomass and less in spores. These are the spores and these are the particles of glomalin. So the how soil particles are intermingled each other and stability is increased. Aggregation is also increased through the hyphal network, and that is that contains more glomalin compound as compared to the spore. So in this case, we have a mycorrhiza. It produces glomalin. It has a potential up to 30% of carbon, soil organic carbon ratio point of view. It can, survive up to 42 years. It is a longer resident time. It's published by one mainly Michaelis Relic group in Montana State University, and after that, India a lot of work has started, and it enhances the soil carbon sequestration, reduce CO2 emission into the atmosphere. And how we have gone ahead? First, we have developed the inoculum by raising trap plants, or the same inoculum which you have identified from the long-term field trials, and then we studied how these mycorrhizal fungi can help the carbon sequestration. We have also targeted one long-term field trial to compare and then calculate the total glomalin to soil organic carbon ratio, and after the extraction of glomalin, what was the residue organic carbon left after the glomalin extraction. We identified the practice of the glomalin, the soil carbon sequestration, which enhances the soil carbon sequestration in soybean maize system. So this was the soybean maize system we have developed under the National Program on Soil Carbon Sequestration, and then we also compared with the long-term field trial, and that was the soybean wheat and soybean chickpea. And then we compared both after the inoculation of mycorrhiza and as well as the native mycorrhiza which is being multiplied under the long-term field trial. And then we studied the carbon pools, the total glomalin content, glomalin stocks, carbon stocks, and total carbon stocks. And then we calculated the soil carbon sequestration. This is the methodology we used. Infectivity potential, we assessed the same protocol, the Gardner-Nicholson. This method is being used to count the spore count, but the infective potential we have published long back. It is being used globally as well as in our biofertilizer companies. We also assessed root colonization, glomalin extraction, the Wright and Upadhyay method, and of course soil organic biomass carbon and CO2 respiration all. These are the different treatments of glomalin extract. You see, in the organic applied plots, the color of the extract is dark and very bulky. if you feel, you will find that very bulky than the mercury and very sticky, 300% higher sticky than the Fevicol, this glomalin. And you see, imagine how it is acting as a cementing agent for the soil particles or in the aggregation. The range of residual organic carbon was negligible after the extraction of glomalin, almost 2.2 to 0.3% left, and the removal of or the loss of carbon might have occurred during the washing, because the glomalin extraction we extracted through at higher temperature, 120 degrees centigrade in the autoclave. Or the pressure cooker method is also being used. So during this treatment, harsh treatment, some protein is lost. The ratio of glomalin to SOC was found to be highest in soybean maize intercropping, and that too under organic, and found to be the range of 15 to 80%, 18%. Although up to 30% was reported, but we reported 18%. But from India, up to 16% reports are available. But we done some innovation during the extraction process. So in this case, you see, these are the different treatments under the DST program. Wherever organic and mycorrhiza and the maize system is there, the carbon stocks is also higher and the glomalin stock is also higher. So this indicates that maize and organic practice is favoring more mycorrhiza glomalin production as well as carbon stocks. So that is indirectly helping the plant or the system to sequester more, and also at the same time increase the crop productivity. Whereas in case of soybean plus maize, same we observed. But in case of inorganic, the carbon stock is less and the glomalin stock is also less. So the AM inoculation in soybean maize intercropping under organic practice had highest carbon stocks. Irrespective of cropping sequence, organic practice with AMF, mycorrhiza, enhances the carbon stocks as compared to without mycorrhiza in organic practice. In case of long-term trial, it's very interesting to tell you there is no mycorrhiza. Whatever resident mycorrhiza is there is multiplying through the practice. So in this case, this is 15-year long-term trial, soybean wheat and soybean chickpea. Wherever 100% is there, slight increase in glomalin stocks. In case of integrated, the increase is more. So it means the integrated practice, inorganic plus organic, is also favoring the mycorrhiza production, higher mycorrhiza production, as well as maintaining higher carbon stocks. But at the same time, if you see soybean chickpea, the 100% organic has more carbon stocks as well as the glomalin stocks. So this practice is becoming more useful. But farmers will not follow because it is not cost economic. Because 15 years following the same practice, then it will become sustainable. And no chemical fertilizers, no pesticides being applied in this trial. So the soybean chickpea under organic practice had highest glomalin stocks as compared to other combinations. In case of the other parameters, the glomalin and CO2 respiration, when the glomalin is higher, CO2 respiration is lower. So there is a negative correlation that has to be there. So that is our main objective, that how to reduce the CO2 emission in the environment. So if you see here, soybean plus maize, the mycorrhiza under organic practice, the CO2 respiration is lower, whereas the glomalin production is more. So that has been, we have reported, and 2 papers are in process under revision and high impact journals. One is in SBB, other one is EJS, European Journal of Soil Biology. Soybean wheat, in case of the long-term trials, the situation in the CO2 respiration is the same, where the 100% organic, where you have more glomalin, the CO2 respiration is less. So the role of organic practice, harnessing more benefit of this mycorrhizal fungi, and it can help the higher sequestration and reduce the CO2 emission in the environment. Although we have not have the system like phase experiment or open top chamber where we can simulate these practice, then definitely we can conclude that definitely at what level of CO2 this mycorrhiza production is more and the CO2 response is less. But at the ambient level, this is the picture that for if the farmer using 100% organic under soybean chickpea, it will sustain the crop productivity as well as reduce the CO2 environment in the system. Overall productivity of the system in general, irrespective of crop sequence, the higher granulate we observed in the mycorrhiza inoculated plots. However, comparatively higher return was observed in the intercropping system followed by sol maize. So it means intercropping is must. And in USDA, 20 years back, a lot of work has been carried out that maize, if you include with cotton or soybean, it becomes more sustainable and can sustain the crop productivity on long run. We also observed here in our system also. So after that, I would like to highlight the use of phospholipid profiling that we have in our laboratory. And this system is very much versatile in the sense that it tells you the living snapshot of a soil as a soil quality parameter. In US, the PLFA method has become a soil health parameter. But in our country, the soil health card, where NPK is the maximum we can do. And if we can sustain that, it's much pretty good. But the PLFA is beyond our imagination. But if we can include, definitely it will tell you that it will tell you everything. It's an impression of overall soil health, because there is a minimum chance of manipulation in this case. Because we are not telling, the machine is telling at what time sample has run and what is the situation of different biomarkers. So the same trial we have targeted the soil health point parameter, that how the CO2 respiration, the glomalin, as well as the PLFA is taking place under this system. As I told you, the organic system is more beneficial in terms of glomalin and CO2 respiration. Let's see in the PLFA. In this case, you see here the more AMF, the blue bar, you see more in organic, and the sol maize is less. In sol maize, you see here not significantly higher, but in case of soybean, it's much higher than sol maize in case of organic practice. I'm targeting here on mycorrhiza, but the total PLF you see here is not very much different. But the organic practice certainly favoring higher PLFA, and that indirectly total microbial biomass that already been observed in the earlier slides. So here also the same thing we observed. So in case of intercropping, organic practice is favoring more AMF biomass, significantly higher. So that built you confidence that this practice, it is not only enhancing glomalin, reducing CO2 environment, but also improving the total overall biomass of AMF fungi. And this paper is under revision. Hopefully in couple of weeks, it will be, appeared. Let's see. In case of long-term trial, where we not inoculate any mycorrhiza, whatever resident mycorrhiza is there, it is being multiplied. Organic practice, total PLF is higher. It means higher microbial biomass is there. Mycorrhiza is also higher, which is 16-1 omega 5, which is a PLFA signature for AMF biomass. And the NLFA also we have observed, but in this case, I am showing you the total PLFA for all these biomarkers. So it's very interesting to tell you that this system, long-term system, is not only increasing the glomalin as well as the reducing the CO2, but also improving the overall soil health, including the mycorrhiza. So then after how to produce, because we have a mycorrhiza, we have identified the practice also, then how to produce. Lot of companies are being supplied these mycorrhizal fungi in the market. Quality is questionable, I told you earlier also. So, but major problem is with this mycorrhizal fungi is a obligate symbiote. The pot culture method at present is largely we use, but the other method like root organ culture, where you use the agrobacterium rhizogenes transformed root, is also becoming more popular. Most of commercial companies, they produce mycorrhiza under the root organ culture because hairy root culture are being used. But again, the adaptability of strain is questionable. The spores may not germinate, although we don't find any report that the in vitro produced mycorrhizal fungi not germinating in the field. But why the farmers are not taking, that is questionable. If a product is good, then definitely, and farmer is happy and he is getting more yield, he will again and again he will ask the same product. So that is the beauty of the microbial, any microbial product. So first we have to develop a starter culture. That how to develop a monosporal culture. We can use the microtip. You can just 1 or 2 gram fine sand used and small seed of like a lucerne or a medicago, small seed, these grass, the fodder grass seed can be used as a host plant. The small seed I am telling you because the size of the microtip is very small. So if you use the sorghum, the size of the seed is bigger. So better avoid that and use this lucerne or any other plant which is small in size. Seed size should be very small. You can also use the slide technique where you can use a single spore, and then after that you can go for microtip and then bigger size of pot. This is how you can develop a starter culture, and then you can use the pot culture method and then farmers on farm production method. This we have optimized in our conditions. This method is very popular in the sense that if you have a starter culture, you can give it to farmer. He can produce at his own site using the cycle of the different tap plants. Like in this case, we use marigold, sorghum, methi, 3 hosts we have used, and then you can also use sorghum or maize in rotation routinely. So the farmer can not only use for production purpose, he can also produce good seedlings, well colonized seedling like tomato seedlings. He can colonize, well colonized with mycorrhiza, and he can use for transplantation purpose. And below ground, he can use as an inoculum for the application in other crops. So these are some of the benefits. It is highly adaptive for application agriculture crops. Technology is suitable for a small, medium-sized enterprise. We can meet requirement of FCO. As you see the fertilizer control order 1985, it tells that 60 spores per gram and 1200 infective or IP infective potential has to be there. So that can easily be attained through this technology. The FCO frequently make amendments in all the biofertilizers, but by and large, this is the situation that 60 spores per gram and 1200 IP per gram. This method is less expensive. This system can be used for production of transplanted crops. Like I told you that tomato, chili, all can be used. So the farmer have a win-win situation. He is selling the good seedlings, healthy seedlings, at the same time producing the mycorrhizal inoculum also. The mycorrhiza, it is better than other bacterial biofertilizers in the sense that it can survive up to 50 degrees centigrade. As compared to other bacterial biofertilizer, any commercial packet, the moisture percent should be much less, should not be more than 10%. Ideally is 5 to 8%. Whereas in case of bacterial biofertilizers, it is more than 40%, which is recommended for carrier-based biofertilizers. Liquid biofertilizer is different. That is even more beneficial if we can use for bacterial biofertilizer. But for mycorrhiza purpose, the granular formulation having 5 to 8% moisture is optimum. And other method is root organ culture system. As I told you, this is a more skillful method. You have to use hairy root culture using aggregative rhizogenes. This dual culture require the multiplication of both simultaneously, the roots as well as the mycorrhiza. More roots are there, more mycorrhiza are there. Then you can produce mycorrhiza. You can also make formulation, root beads or shared roots or granular formulation or whatever, or you can form the tablets also. So the inoculum contains shared roots or the spores plus hyphae and colonized root segment or a cocktail of all. So this is the situation that many commercial companies are producing in the jars. So the method, the quality assessment is very much important. Without quality assessment, we will defeat our bacterial or our microbial cultures in the market. And that is happening, that farmers are not getting benefited. The quality is must. So the quality parameters for AMF mainly includes the assessment of spore count, if the inoculum is a spore formulation, or if the inoculum is a mixed form, then you have to assess both, or the IP you have to assess. This is the infectivity potential assay. You have to grow the inoculum for 14 days, then you can assess the quality using the infective propagules or the infection units formed on the root system. And this method is still being widely used by the commercial companies all over the globe as well as in our country. Although there is a question, if you provide a culture to 10 different scientists, you will have 10 different count. So the microscopic method needs a skillful person. The person who is expert in the microscopy, he can recognize the microbial structures. Otherwise, he may be counting the Olpidium infestation. So I mean that the biochemical or molecular method, there is a need to develop biochemical and molecular method to assess the quality of these microbial fungi. Although industry are not very much interested, I talked to the mycorrhiza editor also. He told that industry may not be happy that if we challenge the method which is currently being followed. But anyway, as a microbiologist, we have to further invent in this area. So this bioassay method is we can assess in our laboratory condition. You can grow sorghum in 100 ml pot for 15 days, and then you can assess the infective potential through the microscopy. Other method, as I told you, the neutral lipid fatty acids, which is 16-1 omega-5, which constitute up to 70% of the NLFA in the spores, and it can be used as AMF signatures. This we have carried out and observed very good result. This is a high throughput method, and in 3 days time we can quantify 96 samples, and this through column chromatography. So apart from neutral lipids and PLFA, the neutral lipid mainly covers the spore count, spore biomass, and the PLFA mainly accounts the hyphal biomass. So if any material is there in the inoculum, mycorrhiza, so if you take 1 gram of soil and we don't know whether it's a spore form or infected roots, so we can assess the total PLFA, means the mycorrhizal PLFA, 16-1 omega-5 PLFA, as well as the 16-1 omega-5 NLFA. So if you total both, you will get the total count of the mycorrhizal biomass of that inoculum. So this the setup we have developed at our place, and through the GC, we can analyze a sample. In one sample takes almost 12 minutes So in total, including everything, it takes 3 days time for 96 samples. And we have observed, and we published also during 2005, that and compared with the microscopic method and the lipid biomass, and the parameters are positively correlated, significantly positively correlated. So therefore, it can be used as one of the parameter or a method for assess the quality of AMF inocula. The sterling fatty acid, which is the total of NLFA and PLFA, we have also assessed. So all the three forms of PLFA, AMF signature PLFA assessed and compared with spore count and AMF colonization. So it was highly positively correlated, and it can be used as a method for AMF assessment. So some of the recognitions we have got from this work: DBT fellowship awarded to work on signature fatty acid, many best paper presentation awards been awarded to my students, and also as far as the take home message and the future line of work, so the studies on root geometry of cultivars should be continued for breeding soybean or any other crop varieties. So that is must. So that will tell you the responsiveness to the plant system and the mycorrhiza. And we have to evaluate the P response curve using the non-linear response, and breeding and evaluate the lines for mycorrhizal dependence under low input conditions. So whatever means variety you are evaluating, the performance has to be evaluated under low input conditions. Then you can really get the benefit of AMF and the performance of that variety or that line. Extraction protocol for higher recovery of glomalin has to be optimized as per the soil type, because in the clay soil we have more humic acid. It is difficult to remove the glomalin. So we have to further see the method. So we have optimized method for black cotton soils, and we have gone up to 10 cycles, whereas as per the method only 3 cycles has been written. So one has to optimize. Easy to work with sandy soils, because only the easy method at 20 millimolar and 30 minutes would be enough to recover the whole glomalin, whereas in case of black cotton soils we require both more than 30 minutes as well as 50 millimolar buffer extract solution has to be used, higher concentration. Mass production of mycorrhizal inoculum, its quality assay and third party evaluation at multi-location need to be strengthened, because we are lacking. If we are developing a product, publishing a good paper, if the product is not taking to the farmers and the farmers are not taking, it means something wrong. So the third party evaluation, somebody else should say, and that is the farmer, that our product is working. So that is all about. And you see the top agricultural industries, there are a lot of potential of microbial products all over the world. See, almost the Bayer, Syngenta, BASF and Dow AgroSciences, this is the major market cap share of these microbial products. So can be used as a lot of potential of these microbial products. And if one is interested to see how to calculate the carbon sequestration, but it's very difficult to see. So this is the one good paper published long back, and the Journal of Climate can help how to assess the calculate the carbon sequestration apart from the glomalin stocks and carbon stocks we have assessed. At the end, I should acknowledge my mentor from USDA, where I learned all these techniques, the glomalin and PLFA, and the DST program for funding the glomalin carbon sequestration, workshop organizing committee, Dr. Kamlesh and other team members. Of course, if I failing my duties, if I forget Professor Anil Prakash ji, who is the convener of this seminar, providing me this opportunity, and also one of my students has been registered under him. He largely worked on glomalin and awarded many poster papers. And Professor B.N. Johri, of course, President Nasi Bhopal chapter, who is really inspiring to all of us for providing and creating this opportunity. And at the end, I acknowledge my director. And these are the ambassadors of my team, because alone we cannot achieve anything. So this is all my students' work. I published as well as just presented for you all. Thank you very much.
Thank you very much, sir, for your this nice and very excellent presentation, because so many new things we learned today from your lecture. And Arpit, I think we have 2-3 questions to pick up for
Yes, sir.
for sir's.
Yes, sir, we have some questions for sir. And the first question we have, sir, biopesticides suitable for vegetables as fruits are generally damaged by the insects or worms.
Yeah, actually, I am not able to hear properly, and I am not very happy with this as compared to Zoom, because I myself, when I am repeatedly speaking, then I am feeling that I am speaking 2 times. I don't know about the audience or the how my voice is going.
No, sir, it was fantastic, sir. The entire presentation was good and informative. There was
please,
no
please
hindrance.
repeat your question again.
Sir, question was, is the biopesticides are suitable for vegetables as fruits are generally damaged by insects or worms?
Yes, it is highly suitable. I tell you one example, the entomopathogenic nematodes, which is one of the most popular biopesticide, which doesn't require any registration, and it not only helps to, kill the nematodes and the insect, white grub and all, which are damaging the root. So this could be one of the good biopesticides which can be used for the vegetables as well as the golf course, because we see the mole cricket and white grub, that is also a big problem. So this could be one of the biopesticide being used globally, but in India it is not very popular because the shelf life is a big problem, the delivery is a big problem of these EPNs.
Thank you so much, sir. The next question we have, which is the most reliable method for determining the organic carbon content of the soil?
Walkley Black method is the only reliable. 1935. Still everybody is using that.
Thank you, sir. The last question we have for you, how to isolate the mycorrhizal fungi from roots and soils?
Use the Gerdman-Nicholson method, wet sieving and decanting method, 1963. You can extract the spores and then you can develop your own culture and multiply using the trap plant or any trap plant. But only thing that you have to use your substrate properly analyzed. It should not contain the pathogen and not high phosphorus.
Thank you so much, sir. That's all from the QA session. Over to you, Dr. Chaure, sir.
Thank you, Arpit. Now I'm handing over the mic to our convener, Professor Anil Prakash, sir, for his concluding remark.
Okay, thank you very much, Kamlesh. And really, I must congratulate you for organizing this workshop on the biofertilizer and biopesticide, new developed novel biofertilizer and biopesticide. So this
Sir,
particular.
Sorry, sir, one
Yeah,
lecture
my voice is
from,
not clear.
No, actually one lecture is left. Dr. Ramakrishna is there. He is going to present actually. Yeah.
Oh, I see. Okay, thank
Yeah.
you. So,
I just like we just like to hear
yeah,
the
because
remark
really,
of Mahavir sir's lecture. Yeah.
okay. So, okay,
Yeah.
thank you. So Dr. Professor Mahavir's lecture is really very informative, and elaborative lecture on the biofertilizer. Particularly, he spoke on the AMF, arbuscular mycorrhiza, and he also given the about that how the carbon sequestration is managed with this AMF. So it is a very good and nice lecture, and we are working together on that. Particularly, one of the students, she is working on this carbon sequestration. So she has worked on very good work, and she has also submitted a PhD thesis on this particular aspect. So with these words, I must say that about that a very good lecture, informative lecture given by Professor Dr. Mahavir Prasad. So now I request Kamlesh to introduce our uh, next speaker, Ramakrishnan. Over to the Kamlesh.
Yeah. Thank you very much, sir, and thank you, Dr. Mahavir Sharma, sir, for giving a very important time to and giving his insightful talk to us. So thank you once again, sir. And now I am inviting our next speaker of today's session, uh, Professor Ramakrishna Usrika. Professor Ramakrishna is working at Department of Biochemistry in Central University of Punjab and also leading as Dean School of Basics and Applied Sciences in Central University of Punjab. Sir has received his education from University of Pune and he was associated with National Chemical Laboratory there as a researcher. He has conducted, he has done MPhil and MSc from University of Hyderabad, Central University of Hyderabad, and he has experienced more than 30 years at various positions. He has published more than 70 research publications in various reputed journals and having citation more than 4,700 and H index of 32. And his research annual conference of Association of Microbiologists, and also he has been chaired so many national and international conferences at various locations, and he has completed so many research research projects which have been awarded or funded from leading leading funding agencies of India and international. He is peer recognition as an editorial board member of journal like Environmental and Analytical Toxicology and Frontiers in Plant Biotechnology. And just a second. And he is having a postdoctoral fellow in his lab and PhD, so many PhD students, and he is having ongoing projects from UGC, CSIR, and DBT, SERB. So I have to read so many things because he is having so many expertise in the area of plant growth promoting bacteria and arbuscular mycorrhizal fungi for enhancement of yield and biofortification of wheat and soil health improvement. And also he has worked on molecular mechanism of multiple stress tolerant plant growth promoting bacteria for enhancing abiotic stress tolerance in various cereal crops. So with this expertise, I would like to invite Professor Ramakrishna sir to enlighten us with his expertise in this particular area. Sir, over to you.
Good afternoon, everyone.
Yes, hello. Sir, may I help you to share your screen?
Yes, yes.
Yes, sir. On right side, you will see there are icon list. You have to click fourth icon from above.
Okay. Fourth icon. The plate. No. 1, 2, 3, 4. This one. No.
Yeah.
This
Yes, sir. Icon is there.
Okay. Share screen. I got it. Okay.
Just click on it. It will ask to install a Google Chrome extension. It will take 15-20 seconds. Yeah.
Okay. Okay.
Just add that extension. It's added, sir. Yeah. Again, click on that. Close it and again click on that entire screen option icon. No, screen share option. Again, click on. Again, first. Yeah. And select entire screen option.
Okay, here. Yeah.
Yeah.
Okay. All right.
Yeah. Yes, sir. Minimize this screen, sir, and then start your presentation slides.
Okay. You can see the slides right now.
Yes,
Yeah,
sir.
perfect.
It is visible, sir.
Okay. Thank you, Dr. Kamlesh and Dr. Anil for giving me this opportunity to talk here, and Dr. Kamlesh for the introduction. So I will start on plant growth promoting bacteria and fungi as biofertilizers. So I will talk specifically about two different sets of experiments or research areas that we performed. So I will not spend much time here because you already had quite a few seminars talking about plant growth promoting bacteria and AMF and how they will be useful. And you can use them in place of or reducing fertilizers. And one thing is, although this seminar is, this workshop is on biofertilizer and biocontrol agents. So these can also be used under what I call multitasking. So not only as biofertilizer, but also you can use it under stress tolerance. As some speakers there talked about salinity, drought tolerance confer, and we can also use this for remediation, like what we call rhizoremediation. That means plant plus microbe together. They are trying to remediate soil which has heavy metals or some other contaminants. So these are good alternatives to do the remediation instead of using the some of the conventional methods which are not environmentally good. So here again you might have got quite a few seminars about plant growth promoting rhizobacteria, or here we call bacteria, because we are also talking about bacteria which are not necessarily sticking to the plant roots. They can also be free living in the soil. So they generally do this growth promotion using direct methods given here, like nitrogen fixation. They produce phytohormones like indolacetic acid, and they help in solubilizing phosphorus, which is present in the soil, mostly in insoluble forms. And then they also produce some substances like siderophores, which will help in ion chelation. Iron is something that is also needed for many pathogenic bacteria, so it can also be a compound that will help in biological control. And they reduce ethylene production by producing the enzyme ACC deaminase. So the other ways is indirect ways is antibiotic production. So this is one thing, antibiotics that will not kill themselves but kill other microbes. And this is also an area that has received a lot of attention. Antibiotics means why the production is good or bad. Are we making the soil with all antibiotics, which we don't need? So there are different arguments for this. We published one review also in this, which I'll show you later. The other things here, they produce antifungal metabolites, compounds, and some enzymes. So those are some of the things they do indirectly. So this one is showing you how the plant growth promoting bacteria and fungi, they help for as biofertilizers and also biocontrol agents. So they produce a lot of compounds. So what we call secondary metabolites and other compounds, which can be primary also, like you are getting organic acids, amino acids and others, which will help for the plants to grow well, and many other functions it has. So this slide is showing you on one side how they can deal with abiotics. stress. Okay, the abiotic stress, like some of the previous speaker, Dr. Sharma talked about AM fungi. So some of the goma species, they can help in drought tolerance, and many bacterial species were also found to do the same. And then salinity, few are given here, but there are many more doing that. And biotic stress is another area where these bacteria can help, bacteria and also the AMF. And one area is where you can use both of them. And even a single bacterial species, if it is able to do both, like abiotic and biotic stress, that will be great. So there are a number of studies where people are trying to find out at the molecular level, what are the pathways, what are the signaling mechanisms that has an overlap between the biotic and abiotic stress. So in the past, in general, people studied abiotic stress separately, biotic stress separately, each kind of the stress separate. But there are a lot of things that happen at the molecular level where you can see an overlapping kind of regulation. If you are able to target those kind of molecules, whatever type molecules they are, and if you are able to use microbes that can give a broad kind of resistance, not only to the biotic, abiotic, that will be really great. Okay, this one is telling you something about ACC deaminase. So a number of these plant growth promoting bacteria produce these. So tryptophan is an amino acid from where we get a lot of indolacetic acid. And one on this side is showing plant cell. So here we are getting the SAM for methionine, and then you produce ACC. And stress creates some kind of imbalance here. So in bacteria, you have ACC deaminase. The ACC can go, can be channeled here, and then you produce alpha ketobutyrate and ammonia. So reducing ethylene in general may be good for the plant, except we know ethylene is good in some areas like fruit ripening. A lot of ethylene is required, and some other developmental program it is required. But in other cases, it's probably good to have less level of that, where actually the PGPB actually help there. Okay, so here we are looking at how this PGPR help in salt tolerance. So here you can see again this bacteria can produce a lot of antioxidant enzymes which are given here, and they can produce a lot of osmolytes, ACC deaminase, we talked about it, and these can get also into the plant cell, and they help in reducing the reactive oxygen species, and in general osmoprotection they provide. So they are helpful in plants in a number of ways. And one of the things when you have a lot of salinity or this thing is you want to reduce, maintain the ionic balance. So a lot of sodium can be there, which actually plant is taking up. Then you have a higher sodium by potassium ratio. So PGPB can help in reducing that that particular ratio by using a number of mechanisms. So this is talking about the AMF, what they do in salt stress amelioration. So this again might have been covered in some of the previous talks here. So they actually improve. They produce antioxidants, improve the water use efficiency, and all those things under conditions where you have a lot of salt stress, and then the plant productivity is going down, your photosynthesis is getting reduced or almost shut down. So there they really help to keep the activity of the plants to a level where they can function in somewhat normal level. Okay, here we are talking about again AMF, so I won't go here again because again Dr. Sharma talked a lot about this. So how these AMF can go inside the cells, and they can spread there, and they can give help to the plants, and plants also help them. Here we are also talking not only about AMF, but also we are talking about bacteria. So together, what they do and how they work as a symbiotic association. So you have a symbiotic association between PGPR and plants, and the same way between AMF and plants. So this is a tripartite kind of symbiotic association. Okay, so here we have different kinds of abiotic stresses, and this actually can help in, can happen in nature. Okay, so here we are talking about different hormones, those are produced, and how they can influence. So microbes produce different hormones, and they can reduce the negative kind of physiological and molecular responses in a plant. So which are put here on this side. You can see excess ROS is produced. You have nutrition disorder, hormonal imbalance, photosynthesis is going down. So all these can be counteracted to some extent or even to a large extent by microbes in the soil. Not only soil, those which are associated with the rhizosphere, with the plants here, and also those which are endophytes, they can also help. So overall, they are helping here, and in nature, what happens? You may not have just one kind of abiotic stress. So here we are showing here 5 different ones. Obviously, you won't have heat and cold at the same time, but you can have heat and drought at the same time, or some other combinations also are possible with heavy metals or salt. So how plants deal with more than one abiotic stress is the key, and bacteria that can help, or microbes, those can help these processes, will definitely increase agricultural productivity and productivity of many other plants. So you have seen a number of people do these omics technologies. What we mean is like transcriptomics, metabolomics, or proteomics. So we treat the plants with PGPB or AM fungi, and then we are trying to isolate from the plants different RNAs, and then you are looking under control and treated conditions what happens at the gene expression level, at the level of say microRNAs which are regulating the genes. We are looking at different metabolites and proteins. So what this is telling you is which genes are differentially regulated, the same way proteins and metabolites. And then we can study how they are interacting with each other. Like proteins can interact with say different metabolites and even with each other. So if you are able to look at that, it gives us an idea about what are the metabolic pathways those are affected in a plant, and what are the physiological parameters those are changing that are accompanied with the molecular pathways. So once we get an idea, we can also probably get some kind of biomarkers which are changing, although it is not easy to get that, but okay, specific metabolite is very highly upregulated, or it is not present, but it is coming because you are putting these bacterial fungi. So this is one area where we want to look at, and then we can target those particular genes by using say gene editing and pathway engineering. So you can get a genetically engineered plant or a PGPB or both. But of course in India we are not allowed to use genetically engineered plants when it comes to say food crops and all. But we do use some other plants like which we don't eat like cotton, or some other plants that can have some commercial value but not a food kind of value. There we may be able to use. Although again genetically engineered bacteria can create a lot of problems, especially if you are using them in soil. But of course we can use it in the lab and do a lot of manipulations. In the soil, the problem is you are putting some genetically engineered bacteria, and how it interacts with the native bacteria, how it will affect in the short run, and more important in the long run is the key to looking at all those. Unless we have an idea of what happens there, it is probably not a very good idea to introduce genetically engineered bacteria into the soil. Okay, this is one way. The other way is we don't want to use a transgenic approach. So you can use a non-transgenic approach. You can look at cultivars or land races, okay, and where you can see these particular metabolites or proteins, whatever you are seeing at high level or low level, they are naturally present that way. So those may be useful for us to use naturally without creating any transgenic plants or bacteria. Of course, it needs a lot of screening experiments, which probably those working with agricultural crops or non-agricultural or IIA, Indian Agriculture Research Institute, those kind of places probably better done. Okay, so these are two of our that we published recently. One is in Applied Soil Ecology and the other one is Science of the Total Environment. So in the second case, we are talking about marginal soil. So marginal soil is one big topic where we cannot really grow. Like normal growth, it may not work. So we can use different kinds of bacteria for remediation and even growing plants for good product to use, maybe not for agricultural, but non-agricultural kind of uses, we can use that. Now I go to my work here. So the first one I will talk is the work that we did long back when I was working in the US at Michigan Tech University. So here what we did is we got some soil samples. So actually what we call this is lake sediments. Lake sediments at different levels, we got some samples, and from there we isolated copper resistant bacteria. So let me give you something here. The lower bullet point is saying history of copper mining. So in that region there was copper mining that was done for several decades, and at one particular region they were doing copper, they're mining and they're throwing it in a lake there. So it will go down and settle at the bottom of the lake where you have different layers. So one particular layer you see that corresponds to the region when the copper mining took place. So we took that kind of layer. For example, these 3 to 3.5, 6 to 6.5 centimeter. Those are the regions where these sediments had high levels of copper. So that we got, we suspended in some buffer, we put it on plates to isolate copper resistant bacteria. So at that point, my interest was just to isolate copper resistant bacteria and maybe use them for some remediation viewpoint using bacteria. Okay, then we identified these organisms. They were found to be Pseudomonas species they belong to. And then we thought, okay, let us look at some of the properties. We looked at all the properties resistant to different metals, phosphate-solubilization, indole acetic acid. So once we did all this, then we thought, okay, let's look at whether they have plant growth promotion when we actually use it on plants. So these are some of the plant growth promoting attributes that we are looking in the lab, which may or may not translate into plant growth promotion when you are using actually with plants. So these have been characterized with IAA production, soluble phosphate, and all these heavy metals here. So based on that, we selected some strains, and we used maize and sunflower to start with and see how it will help. Okay, so we used different ways to inoculate these bacteria. So here we see, this is in the pod study. So you have A, where you have normal soil plus bacteria. So I want you to draw your attention to the D. D is only copper is there. This is artificially inoculated. So you can see the growth has come down. When we put bacteria, C, is bacteria plus copper, it comes back almost to the normal level. So this clearly shows to you how effective it is. Okay, then we did all these other assays that tells how this one is working. So I won't spend here too much time here. I will just go through quickly. And interestingly, when we use the same kind of conditions for sunflower, it did not really have much effect, putting that much copper as we put for maize. So sunflower is supposed to be more resistant to this particular heavy metal here, that copper. So therefore, it didn't have much effect to start with. Then we tried to figure out how much copper is being taken by maize. So if you had only copper and you put bacteria, it takes more than double. Okay, now if you use sunflower, although its growth was not increased, but to take per gram of the plant, here also the copper uptake has increased almost to double. Okay, then we did some metabolomic and proteomic analysis on a different set of samples. Okay, so this is another example here we did is we picked up some sand site soil from here, which is heavily contaminated. So you can see the organic matter is very low, total P is low, copper is very, very high. So hardly any plant will grow with this kind of copper. So we used here 4 different ways to inoculate plants to see which one will work better. Out of these, of course, some of them may be more applicable in a field kind of situation. Inoculating, this will work, coating will work, but this probably won't work. The 4th one, soaking the roots and all, on a higher level, if you are doing in the field where you are doing thousands of that, this may not be very practical. So we are looking at these parameters: maize growth, copper uptake, and soil enzyme activities. So what happens to soil health? Okay, so here you look at C2. C2 is the one that is the soil where the plant is growing without any bacteria. So once you put the bacteria, the dry weight increases quite a bit. And here copper uptake also increases. So different methods give different results. And here we saw soil dehydrogenase activity, invertase and catalase. So dehydrogenase and invertase, they generally increase a lot when we put the bacteria there. Then we looked at antioxidant enzyme activity. So N plus N is maize plus normal soil. This is normal soil and TGPV. This is the stamp sand, and you can see here this is with PGP. So you can see the activity. So what do you expect? Generally, we talked about TGPV. Generally, they will increase the antioxidant levels of the enzyme. So here, what is happening in the stamp sand? It is actually decreasing. So decreasing significantly. So we did not understand this result. What is going on here? So in a general level, so if you look at this one, for example, it is increasing. Normal soil, it is increasing, but stamp soil, it is decreasing. Because the levels are so high, the plant is heavily stressed. So this thing we are thinking is maybe the levels are coming back to somewhat normal by putting this TGP. Okay, then these are some of the other papers that we published related to that, where we looked in sorghum, the similar kind of thing. Okay, we did proteomic and metabolomic analysis. Now I'll come back to this study. So this study was done here in India, in my place where in Central University of Punjab, Batinda. So here we isolated soil from different soil places we got from Batinda. We also got some soil from Aizawl, Mizoram. How I got this? Because I went there for some conference 2 years back. So I collected some soil from there, and that also we are screening. So we screened all these first phosphate-solubilizing bacteria screened, and then we looked for IA production, heavy metal tolerance, salt tolerance, all these things. And based on these characteristics, we identified some bacteria here which showed higher indolestic acid production, phosphate solubilization ability, and also salt tolerance based on its ability to grow on plates that had salt. Okay, these are some more. And then we sequence this. When we sequence this, we found that most of them belong to Bacillus species. Some also belong to Serratia, but mostly Bacillus. Okay, then we did a greenhouse study. So this is actually not exactly a typical greenhouse study, but it's an open kind of polyhouse kind of study. So here we actually initially we did used 3 different wheat cultivars. Then we reduced it to 2 of them. These we got from Punjab Agriculture University that they generally used for their field trials, and some of the farmers also in the local area they use these cultivars. So we used different bacterial isolates. So these AHP are the ones that I got from Mizoram. Azole ones. These CP are the ones we got from some cotton soil. These others, this is another one that we got from the local soil, RA6. A is also again we got from Azole, and PR29 and 30 I got from Dr. Ram Teke from Allahabad. So they isolated some of these in that Sam Higginbottom University. So from there I got these to use some of the native versus non-native bacteria. So here what we are doing is not only in bacteria, we also used a combination with arbuscular mycorrhizal fungi to separately and with combination what happens. So these are the greenhouse studies. So here we are looking at total 1000 grain weight. So what happens to that one? So you can see here these are our control, which actually 3086 and 2967. So you can see there is definitely an increase with a number of treatments. You can see CP4, it increases, and when you combine it with the AMF, there is a further increase there. So you can see here the 1000 grain increased like 40% or 35% in that range. And then we also looked at number of grains per spike, and we looked at grain nitrogen content. So here also we got a significant increase in both of them. And then we looked at wheat grain protein content. This is, however, based on the nitrogen content. So there is a formula that is generally used to calculate these. So here you can see how the control is how much it has gone up.